Jacobs with the following title. Okay, Byron, if you could introduce yourself first and then start your presentation. Thank you.
Perfect. Thank you, Vivian. I'm not sure if you can see me. I'm stood across a few devices here, so I don't have one device that does everything sufficiently well. So I'm just going to say hello so you can see my face. I'm Prof. Byron Jacobs in the School of Mathematics and Applied Maths at UJ, and I'm very happy to be here. So I try to get a kind of catchy title, or one that might spark some discussion, and so it's a little bit tongue-in-cheek, but hopefully it'll make sense at the end. So my research is kind of split, has always been split between applied mathematics and computer science. I've always had a strong kind of interest and fascination with image processing, and always a strong interest, fascination, and something of an aptitude for differential equations. And I was fortunate enough to be able to kind of combine those two things in my research, and it's been kind of very interesting and very rewarding. So differential equations are a hammer. I'll start with a quote by Abraham Maslow of Maslow's hierarchy of needs fame. Although I believe he is not the first person to have said this, but he is attributed to saying it in this form. So he says, I suppose it is tempting if the only tool you have is a hammer to treat everything as if it were a nail. We get a little chuckle out of this. I think it's pretty funny, and I think it's pretty apt in so many cases. And as an applied mathematician, especially one that's trained in differential equations, you tend to try and cast every problem you see as a problem in differential equations, and that's kind of where I am. That's why I think about it. So taking a step away from differential equations for the time being, I'm going to give you a very short primer in image processing, just kind of trying to give you enough context for how images exist in computers, and also kind of traditional approaches to processing them. So what you can do, what kind of operations you can do on images. So I think the first thing to note is that the images are represented in a computer as just a matrix. So a grayscale image would just be an n by m matrix. So that'll be a rectangular image with n rows and m columns, or just a matrix, and they're equivalent. There are some technicalities about what those numbers should be in terms of what you can represent on a screen. So the entries in this matrix should all be unsigned integers, so numbers going from 0 to 255, and that represents an intensity of that pixel at a particular point. It's often more useful just to rescale that into a real number between 0 and 1, and then essentially let the computer deal with transforming it back into an integer when it needs to show it to you. We can then get a tensor. So we have, instead of having just a scalar at every entry of the matrix, we can replace the scalars with a tensor, or a 1 by 3 vector, and that represents an intensity in each of the channels R, G, and B. So a red intensity, a green intensity, and a blue intensity. And then I'll mention it briefly because the DE methods that I talk about later generalize nicely to hyperspectral images, but you can also then have hyperspectral images. We have an n by m picture, and each pixel in that image is a vector of length p. And that would represent an intensity of information at a specific frequency band. And that's used in, I won't try not to get into it too much, but it's got numerous applications, in astronomy to geophysics, and plenty of others. Medical imaging, another big one. And so this allows us to kind of re-envision any numerical data really to be interpreted as an image. So if we have any kind of data, we can kind of cast it as an image by reshaping that data into one of these forms, one form or another. The important thing about image data is that your spatial information is correlated, right? So the pixels next to each other, either up or down, are related to one another, and that forms kind of pictures as we send them. And then sometimes, not always, but sometimes recasting a traditional problem can result in novel solutions, right? So taking a problem in, say, image processing and then cast it as a differential equation problem might give us an interesting solution or a new solution, and vice versa. So essentially, that same idea of looking at maybe screws and staples and things and treating them like as if they were nails, maybe you're going to get at least maybe not a good result, but at least a near result. So just an example here, I've generated a random matrix, so the entries are just between 0 and 1. And if I visualize that as an image, that's what it looks like. So we have pixels with higher values or values close to 1 are represented close to white. And then pixels with values close to 0 are represented as black, close to black. I can do the same thing. I can generate a tensor. So each pixel here is an RGB. So if I take the top left vector here, I can see the red channel has a very small number. Others two are 5.5.6, and that gives us this bluey green. So the red channel is small, but the green and blue are mixed equally, so we end up with this kind of blue-green color. And so, yeah, we can generate color images in the same way, where each pixel is a vector of length 3. You can also think of a RGB image as being like kind of 3 matrices like this stacked on top of each other in kind of the depth direction. So you can treat them individually rather than treating the pixels as vectors. But nevertheless, just to give you, that's just really to give you an idea of what or how images look in computer. So traditional image processing. So these are kind of axiomatic or kind of fundamental tools that are used in image processing, and they come in pretty useful. Maybe not for achieving a goal or solving a problem directly, but certainly in conjunction with other techniques, these things are often super helpful for achieving a specific result. So I've just got this checkerboard pattern that's clearly a binary image, so black and white only, so there's no gray or anything, there's no colors. And these kind of operations are called morphological operators. And what they do is that they operate on the morphology of the image, so they can kind of like modify the morphology. And so we can take a kernel, something like this, so a disk matrix. So, it's got a black background, it's a white circle, and this is of dimension 50, so that's why it looks kind of circular. But I'm going to use one that's small, so it's kind of it has dimension 3. So I think it has a radius of 3 pixels. And what I can do is I can dilate this image, and what it means is that I essentially dilate all the white space with this kernel. So you can think of it as a physical process where I run this kind of white circle in all the white space, and that where the center of that circle hits a black line, it stops. And everywhere where that circle overlaps onto the black space, it kind of eats away into the black space. And so as an overview, I mean, there's kind of an application of this is if you have any white space that you want to be made bigger. So say you've binarized an image and you have that your white space is almost what you want, but it's kind of thin. You can kind of dilate your image and kind of fatten up that morphology, and you can choose a particular kernel. So disk matrix is normally nice because it's symmetric, and you can kind of open up that white space by dilating. Kind of the opposite of that is erosion, where you essentially erode away the white space, and a similar process that happens with actual like mechanical process of operating which pixels should be turned black and which should stay white. And so you can think of erosion, you can either think of it as eroding away some of the white space or dilating the black space. They are actually equivalent. Then you have an opening, which what it'll do is it'll perform a dilation and then an erosion in that order, and the closing performs the opposite. And what you get is kind of this artifact. In this case, you have these rounded edges at the meeting points of these things. So the closing, for instance, is a nice example of what might be helpful. if you wanted to take your input image, right, and you wanted to make each of these squares distinct from one another. So you can actually close the image and kind of eat away at the corners, and then each of these little items is separated. So you think about if you were to pour water into a white space, and these black spaces were like columns of something, in this case your water would basically be able to flow through this image. So kind of the next step, or next kind of natural choice natural step in processing images is using a kernel. So taking this kind of idea of kernel, so it's not a binary kernel in this case, something like a Gaussian matrix in this example, where we have a peak right in the middle, so our highest value in the middle, and then it tapers off as a Gaussian, so it's an exponential squared. So this is kind of the bell curve in 3 dimensions if we're looking at it from the top. And what we can do is we can slide this kernel. I'm going to use a smaller kernel, so it's of size 3. I'm going to slide this kernel all over my image, and I can do it iteratively. So the result I get from doing it once looks like this. So it's that same image slightly blurred. But as I kind of increment the number of times I do it, you can see we achieve this kind of blurring effect. So this is called Gaussian blur. And if you ever played around in Photoshop, or I think even Paint has it now, you can apply effects to photographs, and you can apply a Gaussian blur, and that can achieve a few results. You can obfuscate a background. You can blur some information that you don't want in the image anymore. You can achieve kind of motion blur effects if you do it in one direction. But what's interesting is that you have this linear process. So the process of applying this kernel is a linear one, and you can just slide this kernel over your image iteratively and achieve this kind of blurring effect. What's cool is we can actually choose any kernel. So here I've got a 3x3 kernel. This matrix is my kernel, and essentially is just calculating a gradient. It's a gradient in the x direction. And as you can see, what it does is it's black everywhere, and it's white where we have vertical edges in the image. And this idea of edges and kind of smoothness is an important one. You'll see in the later applications. So yeah, so just by choosing a kernel like this, we can pick out the edges, and there's an edge detector, so it picks up all the vertical edges in the image. And we can transpose this kernel. Simply by transposing the kernel, we can pick up all the horizontal edges. And we can do some combination of those two to get all the edges. And there are many different edge detection techniques. I'm not going to go too much into this, but there's an idea of spectral filtering. So there's an equivalence. I think throughout mathematics, there's an equivalence between kind of spatial representation of functions and spatial representation of problems, and then a spectral representation of that. So kind of decomposing a problem into frequencies. And I think the natural thing that arises is the Fourier representation. So you can decompose any function into a Fourier series. And what we find is when we have, so I'll maybe illustrate this first, and then I'll talk about how it relates to images. But as I increase this point, I'm adding terms to my Fourier series. So I'm trying to approximate the step function. So it starts with a 0 and jumps to 1 when x is 0. And as I increase the number of Fourier modes, right, as I go higher and higher up the spectrum, using increasing wavelength, so going up in frequency, I get a better and better approximation to the step function. And so what you can see is that kind of low order Fourier terms capture slow undulation nicely. So they can capture smooth changes nicely. But when we have these steps, we require lots of high frequency information to capture that, right? And so in images, the stepping is corresponding to exactly these edges, right? So in images, we can actually think of, well, what is high frequency information in images? It's where there is where the spatial variation is very high. So going from white to black and black to white, that's where we're going to get high frequency information. And so it's kind of nice to think about this duality, or this kind of relation between spatial filtering and spectral filtering, where we could apply in the spectral domain just some kind of like low pass filtering. So we cut out all the high frequencies, and we'll get the same kind of result in terms of blurring it, right? So we're going to remove the edges. But it's also important to note that noise is a high frequency, is also high frequency, right? So in terms of denoising, we're caught in this give and take. So if we're trying to denoise an image, which I think is a common thing, or denoise data more importantly, or more generally, we can filter out the noise, but we know in images, edges are also high frequency data, right? So we can't kind of indiscriminately apply some kind of smoothing process to remove noise. I'll show you in the next couple of slides what happens if we do that, because we're going to, yes, we'll filter out noise, but we're also likely to filter out edges or important high frequency information. So that's the kind of end of my very, very brief primer into image processing, traditional image processing. And it's handy if you're kind of delving into this programmatically, looking through a library. And I mean, these image processing libraries have a whole host of kind of built-in functions, and seeing what they do and when, and kind of contrary to my title about just talking about walking around with just a single tool being a hammer, but seeing what else you can put into your toolkit. As I said, the erosion or the dilation in the last example, I'll show you a real world example, and I actually use one of those tools just to kind of like fatten up a binary result that I get. And so it's handy to be aware of what traditional techniques exist, be it in libraries or be it if you want to recode it yourself, just so you have those kind of fundamental tools. So we can treat an image as a discrete initial condition for a well-posed PDE. So what I mean by this is, we know for a PDE we require a few things. We require initial data or initial condition, and when we discretize that PDE, we need to when we discretize that PDE, we can also discretize the initial condition. So why not treat our initial condition as image data and impose upon it the evolution of that image following the dynamics of a PDE. And so here I have given just a simple linear diffusion equation. So D is just a constant, the diffusivity, and then D squared U or del squared U by del X squared, and del squared U by del Y squared. That's just the Laplacian in 2 dimensions. So this would be an example of processing just a grayscale image. And I'll stick that for now. Just using a vector notation, I can generalize this to P dimensions. So then you could apply this heat equation to a hyperspectral image, and it would kind of diffuse in all directions, both in X, Y, so the spatial directions, but also in the Z, so across RGB or across your frequency bands. Again, kind of building this intuition about the relation between high frequency information spatially and high frequency information spectrally, and also what the intuition of the heat equation is doing. We can kind of make an educated guess of what we expect the result of this processing is going to be. So I say here the heat equation diffuses pixel intensity at a rate that's proportional to the curvature. And so what is the curvature? The curvature is just the second derivative. And if we take the second derivative of a function, a smooth function, you're going to measure, or you're going to get a local measure of its bendiness. How curvy is it? So a straight line has a curvature of 0 because it's not curvy at all. And if you had, for instance, an edge, you would have very high curvature. So if you had an edge in your image, that's the point where your curvature is very high. And so in an image, what the diffusion equation is going to do, it's going to diffuse, so it's going to spread out information at a rate that's proportional to local curvature. So in the image where you have local, kind of high local curvature, it's going to correspond to either noise or edges. And it's going to spread that information out, and that's going to result in the blurring of edges. So it was nice that you kind of get this complete view of, you know, approaching this problem from 3 different ways and getting the same result, and having an understanding of why that is. A nice kind of consequence of the equation being linear is that we can numerically solve the heat equation using we can treat the solution as an image convolution. So I can construct a kernel for this equation, and I can just convolve the image with the kernel, and then get the result. I've just realized that actually maybe image convolution is not as common as I might think. So it comes up in machine learning obviously a lot, in convolutional neural networks. So an image convolution is where we take, I'll illustrate it here. So this thing here is just a matrix. I'll show it you in matrix form. So we take this little matrix, right? So it's a 3 by 3 matrix, and we're going to slide it all over the image, right? Everywhere we can. And as we slide it over, we compute the intensity of the pixel at its center point. And yeah, so that's kind of, not kind of, that's exactly what image convolution is. But what's nice is that we can take this kernel, and it represents a linear process. So if we have a linear equation, we can represent it as an image convolution. So I think Dr. Herbst is going to give you a whole day on numerical methods for differential equations. So I won't go too much into detail here. But roughly speaking, I can discretize this equation. I can approximate the temporal derivative and the spatial derivatives, just using a standard finite difference method, so FTCS, forward in time, central in space. And if I assume that d is just 1 and dy and is equal to dx, so if my image is square, so my steps in x direction and y direction are equal, if my image is square, then I can write this numerical scheme as follows, right? And what this here is an explicit update rule, and you can see this is exactly mirrored in my kernel. So we have dx squared on dt minus 4. That's in the center point of my kernel, right? My I plus 1 term and my I minus 1 term appear here. I minus 1 there, and I plus 1 there. J plus 1 over there, and J minus 1 over there. So we have this kind of up, down, left, right kernel living in this matrix here, right? And I can manipulate that over number of steps. So as I increase the number of steps, we see we have this blurring effect. And I've glossed over boundary conditions. You can treat them in a number of ways, but they're not terribly consequential to the result. It's a minor technicality. It'll take too long to explain for not sufficient information. So yeah, that's what it looks like if you apply the heat equation to an image. And you can think of this as well, if you were to say you're running a factory and you had you performing some process and you wanted to know, you had some hot spots and cold spots in your production process. You could take a thermal image of that and then predict how that heat might diffuse. Right? So how long will it take to reach uniform heat? What is that uniform heat going to be? Where are the hot spots? How might we improve kind of cooling in that hot spot? What would happen if we improve cooling in that spot, et cetera, et cetera. So even though I'm kind of being facetious about using differential equations and hammer, the application to image processing is a real one, in that you could take kind of your in this case thermal imaging data, apply some physical process to it, and get a meaningful result that's immediately interpretable. You kind of say, okay, well, after so long, our heat still hasn't kind of averaged out to a constant temperature. It's still pretty like hot and cold. Just as a kind of really driving the nail in, or like really drilling the point, if I take my Gaussian matrix I took before that achieves a blurring effect, and I compare it to what the heat kernel looks like, we can see that there is a strong relation in terms of what the kernels look like, and so we expect that they're achieving the same result. And I guess if you wanted to delve into it, what does the Gaussian matrix look like in terms of e to the minus x squared, and what does the exact solution of the heat equation look like in terms of the spatial component. I think you might find that there's a strong kind of relation between those things, and so it's unsurprising that these results are similar. All right, so what's next? Sorry, I'm using Mathematica for my presentation because it's must start my video. Okay. Yeah, I'm using Mathematica for my presentation because I can do all this kind of manipulation in real time. It's not quite as thick as I might like. So I guess the natural question, I suppose, when we're dealing with this kind of checkerboard image is, well, what if we have a noisy image, like you see down here? I've added noise to it. What happens if we have this image and we want to clean it up? So we want to denoise, we want to smooth the noise out, so we want to diffuse the noise, but we don't want to blur out all these edges. So two authors, Perona and Malik, suggest an anisotropic diffusion, so it's a nonlinear diffusion. And what anisotropic means is that it's non-homogeneous in terms of its diffusivity. So diffusivity is dependent on the image data itself. And so I guess they kind of said, well, we want a diffusion equation that will not diffuse along, it will prefer to diffuse along edges than across edges. Right? So that if I was talking about diffusing in a point kind of where the white and the black squares meet, I'd want to diffuse in the Y direction rather than in the X direction. So rather than going across that edge, I want to diffuse along it. And so they propose this diffusion equation here. So they have the div of the diffusivity, this C function of the grad of U. If this is constant, then this just comes out as being the heat equation. But in this case, it's certainly not constant. And then we take the measure of the gradient of the image, right? So e to the minus a measure of the gradient on some k. So this is kind of the conductance parameter, which you can tune. And what this says, what this function says is, if the gradient is very small, right, or let's say it's very large, then if the gradient is very large, then our diffusivity is very small. So e to the minus something large is going to go to 0 very quickly. And so our diffusivity across a high gradient or across an edge is going to be very small. And then obviously vice versa. So that's going to be maximal when you're kind of diffusing in a very homogeneous zone. So we don't have a lot of like gradients. So I apply a gradient filter to this image just to kind of explicitly show you where are the regions of high gradients. So these white lines show you exactly where there are high gradients. And so this equation is going to hopefully diffuse in regions here where it's, hello? Was there a question? All right, sorry. This equation is hopefully going to diffuse in regions where the gradient is kind of close to black than where it's white. So just as a quick reminder, if I apply just the linear diffusion, this is the result I get. And if I apply this kind of anisotropic diffusion iteratively, you can see the edges are preserved, and the points where you have this kind of stronger noise kind of persist a little bit longer. So if I go back here, you can see, yeah, you get these regions where the noise is certainly being smoothed, but because there's no structure, there's no kind of like hard edge, each iteration kind of smooths more and more that noise away. And then, you know, picking some large time, doing this process sufficiently many times, we end at a kind of a stable result here. And you can see we've corrupted the edges a little bit. I think the image was pretty noisy. I mean, the intensity of this noise is at the same magnitude of the signal, right? So I'm adding random numbers between -0.5 and 0.5. And so the range of that is the same order as the noise. So the signal to noise ratio is pretty high here. And so we achieve a pretty good result. So that's another kind of tool to put into your article, this Perona-Malik filter, and I suspect it'll exist in many libraries. So using this PDE approach as kind of a framework, so now we can kind of construct PDEs. As you saw, we can construct PDEs to exhibit dynamics that we like. So we said, okay, what if we had a diffusion equation that didn't diffuse across edges, but diffused along edges? We can definitely do that. Similarly is the case, we can say, well, what if we had an image such as this one here? You can see this is some handwritten note, and the text that's written on the back of this page is kind of bleeding through. So there's this kind of ink seepage on the back. And for the purposes of maybe doing some optical character recognition or some automated processing of this image, maybe feeding it as to a machine learning algorithm, to kind of read this, we want to clean this image up a bit. And so what we want to do is say, any pixel that's above a certain threshold should go to black, or any pixel that's close enough to black should definitely be black. Any pixel that's close enough to white should certainly be white. And then there's some in the middle whose fate is still to be determined. And so we kind of need to look around. So this region, so the regions of the text on the back of the page are slightly less black than the text in the front. And it's kind of, it's not an easy problem. So what we can do is we can construct a different, so we have a diffusion equation here. Du by dt is just d of the Laplacian of u. And then we add the source term. And the source term, I'll show you its properties now. So again, we just apply the image data as the initial condition. So if we look at the dynamics of the source term, right? So this is a phase plot of the source term here. So this on the x-axis is the pixel intensity, and on the right-hand side, on the y-axis is the result of this function of this source term, and it's parameterized by a. So a is going to be kind of a threshold. And you see, if the pixel intensity is above our threshold, right, then we add energy to it. Oh, you can't see my cursor, right? So in the region of going from a to 1, our source term is going to inject energy into that pixel. So it's going to drive that pixel up to 1. If for whatever reason the pixel value gets above 1, the source term is going to subtract energy from it. So this part of the graph is negative. So we have a stable point at 1, and we have a stable point at 0 in the same way. In the same kind of following the same kind of logic, if our pixel intensity is lower than some threshold, we're going to subtract information energy from it and drive that pixel value down to 0, right? And whether those correspond to black or white is inconsequential. But the point is, we have 3 points here. Pixel values above the threshold get driven to 1. Pixel values below the threshold get driven to 0. And I think the important thing is that it's dynamic. So I can show you actually, I should have included this, but I can show you now quickly. If I binarize this image just using a standard technique, and what that'll do is it'll pick some threshold, and it won't dynamically process this image. It'll just kind of say anything above, say 0.5 gets put to black, everything below gets put to white. You can see I end up with a lot of this noise down at the bottom, right? These pixels are kind of like all this bleed through is still here, and particularly over the text, you have this bleed through, and it makes the reading difficult. So the advantage of using a PDE-based framework is that we actually have a dynamic process. So the fate of one pixel, wanted to say this pixel here next to this text here, isn't determined by only one value. It's not saying is it above or below a threshold. It's saying, well, what does the local information look like? How does it change over time? If we diffuse it out a bit, does it maybe look like it should be in the background, or is it very definitely supposed to be in the foreground? And so we can do this process, and we end up with this result here. So I can show you as we perform successive iterations of this process, we can see that we end up with a lot cleaner of an image than we did with just the binarizing technique. I'll show you again here. I'll put it in here. So particularly a lot of this information here, and the reason this is all fades away is because the diffusion equation, or the diffusion part of the equation, is really spreading this information out. Right? So it's saying that this information here can get spread out because it's not being driven by the source term. And so we can kind of diffuse that away. And once it falls below the threshold, then the source stream also drives it away. So we do, it's not perfect. It's certainly not perfect, but I think we end up with a much cleaner result than we do with using just a standard approach. And if I apply the Primarization approach to it, we end up with a similar result that, you know, Primarization is really just not diffusing across edges, you know, rather than trying to do some clever thing about driving pixel intensities. So I'll briefly mention another application of PDEs in image processing. There's no kind of technicalities here, it's just an inbuilt function. So it's a process called in-painting. And what you do is you provide an image and you provide it a mask. So you say, treat this part of the image as a fluid, right? So this white part of the fluid. And it uses the boundaries of this mask as conditions, and then applies the Navier-Stokes equation. So that's kind of treating it like as I said, like a fluid, and saying, fill in this region with information around it. And it's kind of, you can see here this kind of deep kind of scratch or gouge out of the painting is filled in, and there's kind of no artifacts. It's a very, very convincing reproduction of what's happened. You can maybe see some slight kind of blurring there. But I thought this is a nice application of PDs for image processing. You kind of treat it as, you know, what if this pixel was a vector field and how would that fluid flow? So here's a real world application. So this is doing automatic morphological sperm analysis. So we're looking at sperms. This is a process. So if you're potentially having fertility problems, you can have kind of sperm analyzed and then try and decide, you know, what's wrong or what's going wrong. Is it a motility issue? Is it a morphology issue? What percentage of your sperm are healthy or unhealthy? And so here's an image, and you can see real world images are a lot less kind of clean than kind of these constructed examples. So we have this chromatic aberration at the edges. This is by virtue of the fact that we're magnifying the image to such a high degree. We end up with the light frequency, so RG and B, kind of reflecting and refracting in different ways. And so away from the focal point, which is the center, we end up with this chromatic aberration, which is why it looks blurry around the edges. Nevertheless, we want to basically be able to say, okay, well, where are all the sperm, and are these sperm normal or abnormal? And so here's a kind of taken from a paper saying what is a normal sperm and what is an abnormal sperm. So these kind of, everything that's not normal here is abnormal. So these are real world examples. These are really nice examples of them, and you can see in the picture above that you never really, it's never really super clear which kind of category these things fit into. But there is a classification criteria for a normal sperm. So number one, the morphology has to follow a specific proportion. So its length to width ratio needs to be something specific. So it needs to be, say, I think it's between 4.5 and 6 microns long, and 2.5 to 3.5 microns wide. And so if it's round, then you get a round head or a small head. You know, if it's too long and skinny, it's in one of these other things. That's one condition. That's the morphology of it. Just talking about the shape. And then you have this kind of lighter region. You can see in this schematic. The tip of it is kind of lighter than the rest of it. And I think this is a good example of it here. And that's called the acrosomal region. And basically your acrosomal region must not take up more than 70% of the sperm itself. So if it were possible to construct some kind of technique to automatically measure, say, here's an image, here are all the sperm, this is where all the sperm are. So you find the local positions of these things. Then you measure these things. So you know if my photograph is, say, 2000 by 1000 pixels, then at this magnification, this is so wide. So you need to build up a scale saying that 1 pixel represents so many microns in so many real microns. So you need to relate your pixel scale to your physical scale. Then you measure these sperm, you say, okay, well, it's so wide, so long, is it normal or not normal? Measure this acrosomal region. And so that's exactly what we're going to do. So it turns out not to be an easy problem. So the algorithm, broadly speaking, is going to be to binarize the image, right? So we want to basically do what we just did. We want all the sperm bodies to be black, everything in the background to be white, ideally. So we're going to detect disconnected regions. So we're going to detect blobs. So this is the process called blob detection. It's a relatively simple algorithm. Again, it exists in a lot of image processing libraries. And essentially what this allows us to do is find where these binarized components are connected. So, or for these blobs that are connected, they form distinct kind of entities. And so we'll hopefully go through and count. So this is, call this number 1, call this number 2, number 3, number 4, number 5, number 6, and record where they are in the image. We're going to find that we're going to end up with a lot of noise. So we need to filter the region. So anything that's not sufficiently large, we're going to need to get rid of. Anything that's not sufficiently elliptic, we need to get rid of. So, artifacts or, kind of debris in the microscope slide can be sufficiently large, but it might not be round, or it might be kind of like spiky, so we can throw that stuff away. And we want to remove as many other anomalies as possible. So we want to basically filter out the regions and be left with as kind of optimal blobs as possible. Then we're going to record and report the measurements. So we're going to say the sperms that we have are so long and so wide, and then we can say whether or not they're in the normal range or not. So here's another image. Again, you can see this chromatic aberration at the edges, and we've got a bunch of heads here. So this is an example of an artifact. This is just debris. Here you can see it's pretty large, so the blob detection is going to pick it up, but it's not particularly round or elliptical. Here's another one. I don't know what that is. Probably also some debris, but that might be too round. So hopefully we can filter that out, and then all the rest of it. So yeah, you can really see that and you have these kind of stripes going along left from the top right down. So it really is a messy image. It's not even noisy. It really is just a messy image. Fortunately, we can binarize it. So I've pulled these results out. So all my code for this is in Python. So I've pulled all my code out and want to rewrite it in Mathematica. But we can do some binarization. It turns out because our illumination is fairly uniform, our binarizing process can, an off-the-shelf on a standard one is pretty good. We don't need to use a PDE-based one. So we will use a PDE-based solution later on. So we get these binarizations, and you can't really tell, but you can see that there are a bunch of different regions that are too small, maybe not round enough. So it has picked up this kind of spiky region here. Blobs, you can't tell from the scale of all the colors, but it's picked up about 900 regions. So I'll show you the data frame of this. So you can see it's 958 rows, meaning that it's picked up 958 distinct blobs. And you can see from this data frame that a lot of these areas have a pixel of 1, sometimes pixel 998, 8 pixels, 2 pixels. So we really want to filter this out. So anything that's not sufficiently large, we're going to filter out. Anything that's not sufficiently, doesn't meet some kind of criteria for this measure of, say, eccentricity or convex area. So we have all these measures that come out of our blob detection algorithm, and we can use those things to filter out what we might not need. So we do that, and we end up with 1, 2, 3, 4, 5, 6, 7 distinct things, and there they are. So I'll highlight them. So you see, we've left with this guy here, even though it's not actually a sperm, it's just an artifact. So maybe our filtering could have been a bit better. But there they are. I've highlighted them. Unfortunately, got rid of this one. We know that this isn't sperm there. So it's done. I would say it's done pretty well. I mean, it's got all 6 sperm in the image, and it's got the 1 artifact which it shouldn't have. So I would say it's doing pretty well. And then a report. So it does the major and minor axis length. So I know the scale. So we had to calibrate this camera. So you take a photo of a known length. So you have kind of calibration slides. You take a photo of it and you say, okay, the circle is printed at, say, 3 microns wide. It takes up, say, 300 pixels from left to right. Therefore, 100 pixels means 1 micron. And so I can relate the number of pixels in the width and length of these things to a true length in micrometers. And there's only one, I believe this is the head here, that has normal morphology. So it has its minor ax length, so it's between 2 and 4 microns wide, and between, I think, 3 and 6 microns long, whatever those thresholds were. So that's half the battle. The next battle is attacking this acrosomal region. So I just want to remind you. So we want to look at taking this image. We want to talk about the lighter part of the image to the darker part of the image, and saying that we don't want to do more than 70%. So we're going to perform what I'm calling as a trinarization, and I invented the term only to find out that it had obviously been invented before. So trinarization is exactly what you expect, rather than segmenting an image into 2 regions, black and white. You can segment them into 3. So black, gray, and white. So I think you can maybe see where I'm going with this for the trinarization. Measure the acrosomal region by area, so just count the number of pixels and report that percentage. So these are those subregions that we just detected and just kind of blown up to show you. And we perform this trinarization. I won't go into the details of how we do it, but this is the result we get. You can see this result is really not super clean. We end up with these kind of these disconnected regions, which should surely be cropped. You can see this kind of this fuzzy nature, maybe this is a good example, right? So I can see that this darker region here in the trinarization is certainly over-representing what's in the image. And so what we could do is actually erode this image. So we could erode this darker image, going back to that very first tool that we learned about the dilation and the erosion. We can actually erode away that darkest region there to hopefully get a better representation of that acrosomal region. But nevertheless, we can count the pixels in this one segment and this other segment, and then compare the two and see what is the percentage. And we do that, and we can report that in our data frame. So kind of wrapping up, I think if you have any data, if you ever have any data that has some kind of correlation, I mean, obviously your data will always have correlation, but if you have data that has some kind of spatial correlation or data that you can recast in a way so that the correlations can be represented as spatial information, then you're kind of looking in, then you're really talking about an image. So it's a natural way for us to visualize this, in the sense that our brains kind of interpret images very quickly. We can kind of get a good insight into it. I think Len's talk yesterday about the kind of misinterpreting correlations in data or misrepresenting them is so true. And so if you can kind of leverage how our brains use images to really emphasize that spatial correlation, I think hopefully it's not an abuse of the approach. Images are just tensors, so especially, I mean, when you take a digital image, it really is just a matrix or a tensor, and that gives you a whole host of things that you can do to it. So any mathematical operations you can do to it, and I think as I've shown, the PDEs form actually a pretty natural choice. It doesn't seem so at first, but certainly once you look at it for long enough, PDEs seem to be a pretty natural choice for processing images, because you can, something I didn't touch on in this talk, but it certainly is true. You can construct PDEs to exhibit dynamics that you want. So you don't have to discover PDEs. You can engineer the PDEs to exhibit the properties that you want. And then you can use your set of mathematical tools to go and prove the results about that PDE. So you can prove that your process is going to be stable. You can prove that your result is not going to generate a pixel with intensity greater than 1. You can prove that your numerical method is stable, that your numerical method is accurate. And so I think that's that's really the power of using PDs as a framework. Something that I certainly learned, so using tools from other domains can lead to new and interesting solutions. And so, maybe it's not the worst thing to be walking around with your only hammer being a differential equation. Certainly, I think so. But having seen how in the real world example of doing the sperm morphology analysis, you can't just apply the same thing over and over and over. You do need to draw on a whole host of techniques. And that's why I say, perhaps having a toolbox full of hammers isn't optimal. So thank you very much. I hope this was helpful, and hopefully it'll be a good primer for the workshop problem next week. Are there any questions?
Any questions from the audience, online and in person?
Yes. Good morning.
Yes, yes.
Can you hear me?
Good morning. Yes, sorry, I can. Yes, sorry, I was muted. Yes.
Can you hear me?
Yes, I can hear you. Go ahead.
Okay, good. Thank you so much, Jacob, for the presentation. I would like to know more about the sperm morphology. Is it the same analysis they use when they are doing the IVF, or where do we normally apply this?
Yeah, so that's exactly right. So essentially what they do is they want to get an idea. So this is normally a process done manually. So what fertility doctors want to do is they want to get an idea of how healthy is a patient's sperm, right? Obviously, in terms of reproduction, there are two variables in the equation, and so you need to identify is there a problem with the male or is there a problem with the female. And so part of this is doing an analysis and saying, healthy males have, I think, about 30% not the normal sperm. So we have a very low normal sperm count, and so it's quite cumbersome for doctors to say, let me go through your sperm and look at all these different sperm, identify is it normal, is it abnormal, is it abnormal in a certain way, because that can indicate maybe some underlying condition. And also, as you can see, I mean, these images aren't clear cuts, right? It's not an objective thing. It's a very subjective thing. So you can ask 10 doctors and get potentially 10 different answers. And so kind of the rationale for this is saying, if we can provide an automated procedure that's objective, it's going to give you the same results, that kind of agrees with the consensus of doctors, then I think then that's kind of a really useful tool. But yes, to answer your question shortly, it's a precursor to doing IVF or kind of a process called ICSI.
Thank you.
Thank you. Thanks for your question.
Thank you. Any other questions? If not, let's thank the speaker again. Byron, thanks so much.
Thank you so much, Vivian. I look forward to seeing you guys.
4 minutes again with the next speaker, Prof. Kana. So I hope to see everyone in a couple of minutes. Khanna. He's the director of DSP NRF Center of Excellence for Biomedical TB Research, and is also connected with the University of Witwatersrand. Okay, Prof.
Good. Yeah. So thank you very much, Vivian, for that introduction, and thank you, Ibrahim, for the invitation to present you. I must admit, I was a bit astounded when Ibrahim asked me to speak in the maths department. As a biologist, I usually need a calculator to work out that. So today, I believe my job is to give you some insight into some of the biological problems we face in infectious diseases in society, and lay the challenge to you as to how do you use your computational and mathematics knowledge to actually address key challenges that face society. So as Vivian mentioned, I run a center of excellence for TB research, and I guess in the context of COVID, you would ask, why study anything else? So why is studying TB important? And so I thought I'd give you some data. These are the most recent data from the World Health Organization. And just to remind everyone that TB still remains a global killer. So what you're seeing are these WHO incidence maps, and the dark colors are countries that have more than 500 cases per 100,000. And what's particularly relevant for us is that you'll notice that a fair amount of the burden is carried by Africa and the developing world. Now, if we overlay that and we look at the estimates of new TB, HIV-infected individuals, so people with HIV-associated TB, you see that problem brings the issue really very much close to home. If I toggle between these two slides and you look at which country remains dark, right? It's our country. So South Africa is home to the world's worst TB and HIV epidemic, right? It is still something that people die on prior to COVID based on death certificates. More South Africans die of tuberculosis than any other bacterial infectious disease. And when we think of COVID, actually COVID has had a very dramatic negative effect on tuberculosis. So this is just basically the trend in case notification, right? So the number of, what you're seeing here on the two axes is the number of cases we find in millions and the number of cases we find over time. And as you saw, as we improved diagnostics between 2016 and 2019, we started finding more people. And then suddenly between 2019 and 2020, that plummeted quite substantively. Now, the TB did not disappear, but what actually happened was that people stopped presenting to the health system. A lot of the infrastructure that we had for tuberculosis diagnosis and management was repurposed for COVID. Many people found it difficult to access TB medicines. And so effectively, the drop in case notification isn't that we solved the problem, but actually the problem went and hid in recesses of society that are very difficult to access through the health system. And if you look, and this is the last slide I'll have on this, but I'd really like to drive this point home, because I think we poised at a change in society with how we look at pandemics. But this is modeling from the Stop TB Partnership. And we often think of these public health interventions such as lockdowns in the context of how they affect our lives and how they affect economies, right? But this kind of models for you how public health interventions affect other diseases. So basically what you see here are two graphs, one of incidence and one of mortality, right? And this is basically the incidence and mortality for tuberculosis. And the different lines are, the blue line was the trend we would have had we not had a lockdown and disruption of healthcare services. And what you see with the red line is, well, if we had a 2-month lockdown and restored services after 2 months, what would incidence look like? And then the orange line is a 3-month lockdown, and then restoration of TB services over a period of 10 months. And what I want you to notice is that we never get back to the blue line. No matter what we do, we never get back to the blue line. So I mean, there was good reason to have these public health interventions, but by implementing them, we have damaged other parts of the health system to the point where it's not clear that they're going to recover at all. I mean, if you look at mortality, we expect a huge spike in mortality because of these lost TB cases that don't present to the clinic. And so that's the context for my lecture to you today. But really, I want to overlay what I want to say to you in the context of how TB presents in the clinic, right? So everyone's familiar with tuberculosis, right? You imagine this disease where people are coughing and they get very thin and there's respiratory insufficiency and people start to waste away. but it's actually a much more complicated disease. And in order for us to manage it using new tools, we need to understand the complexity. And so this is it's a transmission couplet, right? How do you get TB? You've got to share air with somebody who's sick, right? And so usually you have this transmission of disease from people who are infected in disease to those who are uninfected. But as with many diseases, sharing air doesn't necessarily mean you're going to get the disease, right? So there are actually multiple outcomes that happen from sharing air. The first is that you can develop what we call latent tuberculosis infection. I'm sure this is a term you've not heard of. You just think of TB as people who are active and sick and coughing. But most people actually carry TB as latent TB, about a quarter of the world. And if I had to sample people in this room, I would say about a third of you probably have latent TB. The bacteria are inside of you and they've gone in a state where you can't find them and they're not making you sick. But if you become immune compromised, if you get sick with something else, if you get diabetes, as you get older, chances are these bacteria are going to emerge again. Right, and there's a term I have, biomarker negative. And biomarkers, just for people who don't know, is usually a blood signature or something that indicates that you have infection or disease. So here the TB bacteria are hiding. We have no idea that you actually have them. The other thing that can happen is you can get infected, but not immediately develop disease. And we can say that you're infected because you're now biomarker positive, right? In most cases, we diagnose people who have this type of infection by doing a skin test, this tuberculin skin test. And then what happens is people can interchange between these two stages. So you can remain latent for your entire life and die of natural causes and not TB. But the TB can come out and go away, make you slightly sick and go away. It's an incredibly complex and dynamic situation. But eventually what happens is a small group of people, roughly 90% of people, progress. They become sick, and this is the TB you're familiar with. It's actually just the tip of a very, very big iceberg of infection that society sits with. And then these people who are infected eventually have to go on to treatment, and we hope that they get cured, right? But often also what happens is we have, and I'm sure you've heard this term, drug resistance. We have the rapid emergence of drug-resistant forms of the bacteria, which make treatment very challenging. And then we have people who just fail treatment, right? So this is actually what the clinical situation looks like when you hear the word tuberculosis. It's not just somebody coughing, presenting in the clinic who is sick. And so I want to highlight for you today 4 key aspects. The first is, how does transmission occur? And where can you bring your skill to provide greater insight and assistance to society in these spaces? How does transmission occur? What do we do with diagnosis, and how can we improve diagnosis, and how can we use sort of imaging algorithms to help us do this? I want to tell you a little bit about the bacterium, because I think some basic knowledge of this is really helpful. Often when I speak to people in an interdisciplinary space, I find that there's a need to have some basic knowledge of biology for you to be able to really cast a deep lens onto a problem. And then finally, to talk about treatment, right? So that's basically our lecture outline for today, these 4 main points. So let's talk about the transmission of TB, right? So I mentioned this transmission couplet to you, right? And so it's really important to understand what contributes to TB transmission. And I'm sure, 2 years ago, if I gave this talk, it would seem a bit diffuse, but today, in the context of COVID, a lot of this, look at this, most of us are wearing masks, right? A lot of this sort of becomes very, very real, right? But there are 5 main things when we think of disease transmission and trying to model it, and trying to model how diseases spread between people and within society. There are these 5 main things, right? The source case, pathology, environment, susceptibility, and the nature of bioaerosols. And I'm going to very briefly touch on some of these. So let's talk about the source case. The source case is someone who's infected, right? And often, I'm sure you guys have heard of this, people are asymptomatically infected with COVID, right? And it's unclear whether you're if you're asymptomatically infected, how many viral particles are you carrying? Are you infectious? And the same thing applies to TB, right? When we look at somebody who's TB disease, do they have a lot of bacteria? Can we culture them? When we look at their sputum, when we do a smear, is it positive? Can you find a lot of bacteria, bacillary load? And if you can, then that person is very likely what we call a high transmitter or a super spreader. And then things like HIV infection status. So, it's interesting to look at what HIV does to TB, right? So what does HIV do, guys? What does it do to your immune system? Someone shout out an answer for me.
Destroys the immune system.
Destroys the immune system, right? And what do you have to do? You take ARVs and you measure CD4 counts, right? So in order for you to mount an immune response to infectious diseases, you need a functional immune system, right? And for tuberculosis, you actually need those CD4 positive T cells because what they do is they start to wall off the bacteria in your lungs. CD4 positive T cells are quite important for that. And they create these big lesions inside the lung that then eventually keep the bacteria within the lung, right? But those lesions then open, and when you cough out bacteria from there. But if you're HIV infected now, and you can't bow those lesions, right? It's very difficult to find bacteria in your sputum. What happens is the bacteria starts spreading everywhere into your body. And so HIV infected people, whilst they present with worse disease, they actually are not, they don't transmit, they don't have a lot of bacteria in terms of being able to transmit, right? Whether you've had previous history of TB, if your lungs are very damaged, you're likely to be coughing up a lot of bacteria. And then things like resistance, drug resistance. If your treatment's not working, right, and you're still harboring a lot of bacteria, then your chances are you're going to be spreading organisms around. And then the ability to aerosolize. So very much like our voice, our ability to cough is very individual. And you can actually find this in these new algorithms that study cough acoustics. And I'll touch on this when I close today. But you can, a cough is an incredibly complex thing from an acoustic nature. Each person's cough is individual. It's very unique. And some people are good spreaders of disease, and some people, just by the nature of how they cough, don't spread so well. Your lung capacity, and then of course things like your behavior. Do you move around? Do you have a lot of social interaction? What is your hand hygiene like? What is your space like? What is your health-seeking behavior like? Then the next thing, pathology. So I mentioned this, whether you have disease that carries a very high bacterial load and has a lot of lung damage affects your ability to transmit TB to others. And then the nature of bioaerosols. So who's heard of the term bioaerosol? Just by show of hands. Nobody? Right? You've heard it, right? So aerosols, and this is again very topical in the context of COVID, right? What is the nature of the contagion? If you recall, in the first 3 or 4 months of the pandemic, was it airborne? Was it droplets? What was it, right? And so the nature of the contagion is incredibly important when we talk about airborne diseases. And in the case of TB, it's bioaerosols, and you cough up fine droplets, and those droplets carry bacteria, and those bacteria then, when encountering the individual, go into the alveolar space. But the size of those organisms, the size of the bacteria, and when a person coughs, right, you imagine this huge cloud of bioaerosols coming out. The direction of each of the bioaerosols, how long they stay in the air, all of those things become incredibly important. And you can model these. So you can actually take a movie of someone coughing, and then you slow that down, and you model the trajectory of all the individual particles. You can even give the person a food colorant, and you can track some of the bioaerosols, right? And then the environment. So things like ventilation, we have all our windows open here, humidity, how close you are to people. By now, I'm sure you're sick of hearing this term social distancing, but how close you are to people, right? And then the idea of rebreathing of air. Let's see how big is this one. Let's see if these windows weren't closed, right? After about 40 minutes, we would be rebreathing each other's air, at least 10% of each other's air, right? So with no air movement and no air circulation, you can actually model how quickly the air becomes infectious and what dose of organisms climb within the air, right? And then the idea of congregate settings, how many people are in a room, schools, churches, classrooms, university lecture theaters. And then finally, the idea of susceptibility to infection, right? So something like COVID, we're all susceptible, right? We haven't found people who are exposed but never get infected. We haven't moved as a society to a point where we're able to do those studies, right? So this is actually incredibly important. How strong is the immune system? What are the inherent genetic factors that provide you with this idea of infection resistance? So when we talk, and I'm sure you've seen these models, because there's a lot of them in literature and even some web interfaces where you can talk about how does a disease spread and how do you model spread. Actually, that spread is modeled considering these 5 factors, right? And I want to give you an example. This is not a very recent paper, but it's one I could understand, and so that's why I decided to show it to you. It's actually a mathematical model that shows how tuberculosis transmission can occur and how it factors in all these different contributors to transmission and infection resistance, right? So you imagine here's an index case, and in this case, the mathematical model says that the index case is in contact with 4 individuals, and how can you model the likelihood of transmission in that particular setting, right? And so there's these 4 factors. You can work out bacterial shedding. And so you look bacterial shedding here in panel A, and over time, so we assume that as a person gets infected, remember what happens? You're walking around, someone coughs up, you walk through the cloud, and now you get some TB bacteria. You're actually infected with a very small number of organisms. So your infectious period is not constant. Yeah, guys, makes sense? First, you start off with a low number of bacteria. Those then grow and reach a certain threshold. We say beyond this threshold, you are infectious. Then you go on to treatment, so your immune system controls the infection and you drop below that. And all of this we model using known data we have in the laboratory for how bacteria would respond to treatment and how bacteria respond in the clinical setting. And we can work out in a very clear mathematical way what the duration of infectiousness would be, right? And then you can also look at now that infectiousness and how it affects your cumulative exposure with contacts, right? So how much bacteria have these contacts seen, and how much have they seen over time? And you can eventually integrate all of this to give you a model that tells you, well, this person who's HIV infected in a household has 3 rooms where people see each other in the evening and in the morning. The chances of infection in this home is X, Y, and Z. You can start to work out what the risk individuals have in a particular setting. Okay, so I've just made some little summary points for you. So the take-home message from this part is TB is a clinically complex disease, and it's driven by ongoing transmission. And transmission can be modeled using various different elements that contribute to the idea of how transmission occurs. And those are the 5 that I've mentioned. And that these models can then be used to study pandemics and infectiousness of individuals within certain spaces in very controlled ways, right? There are things that are sometimes difficult to model, like the prevailing wind conditions over a community and how that affects TB transmission. But you can put in some variables and develop a best fit model, right? So before I go on, I'm going to just pause. Is everyone good? Yeah, I can't see the hands online, but maybe we'll take those at the end. Yeah. Yeah. Okay. So let's talk a little bit about diagnosis, right? And another problem with tuberculosis. So often I was, we were talking on the car on the way here. Is that when you speak to sometimes you speak to people about vaccines, people are concerned, right? And they say, how did this vaccine get developed so quickly, right? And my answer was, the reason we were able to develop COVID vaccines so quickly is because everyone threw money at the problem. And the reason we haven't been able to develop new vaccines for problems like TB and HIV is because these are mainly diseases of the poor, and people don't throw money at the problem. And so TB diagnosis, especially TB diagnosis in Africa, has really suffered from this dramatic lack of support, political support and financial support. And for the longest time, if I told you that we did TB diagnosis with a diagnostic test that was 100 years old, what would you tell me, right? This in a society that has put a man on the moon and now has a manned mission to Mars, right? So it really tells you that in many cases, the problems in infectious diseases is driven by poor finance. And so when we talk about diagnosis, let's just talk about the bug. TB is caused by an organism. I have a slide later on called Mycobacterium tuberculosis. It's a bacterium, slightly different to a virus. And that bug is acid fast. So acid fast means it stains under certain acidic properties, right? And what's important about it when we do diagnosis is that it's present in sputum, right? So if you're sick with TB and you have it in your lungs, you're coughing up all the time, or you go to the clinic and we ask you to cough, those bacteria come out from your lung and they go into the sputum, right? And so it makes for easy collection. And then we stain that sputum and we look for bacteria, right? And so basically that's what it looks like. These are 2 sputum smears shown here at the bottom. This is an old way of doing it, and then we developed these fluorescent images. And so later on, when we talk about integrating image data into diagnostics, these are the kind of data you're going to encounter when you're trying to develop algorithms for diagnosing TB. But what's the problem with this, right? So we've diagnosed TB this way for now 110 years. In fact, the person who first discovered tuberculosis bacteria discovered the bacteria using these stains, right? That's how old these tests are. But they have very poor sensitivity. So you have to have about 10,000 bacteria, then you can find them. So if you're HIV infected, you don't have a lot of bacteria, you're just very sick, you can't cough up, you're going to get misdiagnosed. And the problem is particularly acute in children, right? So those of you that have kids, you know what it's like when you tell your kids, okay, try and cough or try and take medicines. Managing sick kids is incredibly challenging, right? And especially if they're very young, it's very hard to get them to cough. And so the way we diagnose tuberculosis in children is we do what we call a gastric wash. You push a pipe all the way down into the stomach, and then we pull up some of the gastric wash, right? With the idea that the kids have coughed and they've swallowed the cough, right? And so the bugs are in the stomach acid. And so when you have very few organisms like that, it becomes very tricky to try and find them. So it's got poor sensitivity. And then the problem is that it kills the bacteria, right? So when we do the stain, we have to kill the bacteria. So you actually don't know which bacteria are alive and which bacteria are dead. And that's going to become incredibly important for the problem we're going to present to you later on, right? So the idea of seeing which ones are alive and which ones are dead is incredibly important from a diagnostic perspective and from a treatment perspective. Because if you come in with TB and we start giving you drugs, in a week's time, we'd like to take your smear and say, hey, the bugs are disappearing. But if both live ones and dead ones stain, we can't tell whether you're responding to TB treatment or not, right? So that becomes incredibly challenging from a clinical management perspective. And then the idea of performing poorly in individuals like HIV-infected people and kids I've already touched on, right? So thankfully, over the last 15 years or so, we've had some new diagnostics coming, right? So the other thing we do is we culture. So culture is, can we grow out the bacteria, right? So this is also a very important question with COVID, right? So if you cough out organisms, how long do they live in the air, right? I mean, do they live for days? Do they live for weeks? So do you have to keep cleaning spaces? Do you have to keep fogging rooms? Do you have to, a lot of this is unknown, I think, in COVID, and it's even more unknown for tuberculosis, right? So the gold standard for TB diagnosis is this thing we call culture, right? And so basically what it is, it's this automated system where we take your sputum, if you present in the clinic, we decontaminate it and we add it to these tubes. And we say, can the tube sustain the growth of TB bacteria? And just as a bit of trivia, it's quite a neat system. It's called MIGIT, Mycobacterial Growth Indicator Tube, Mycobacteria, TB bacteria. And so it's got a bit of media, and at the bottom, it's got this little fluorescent dye. And the dye is turned off, right? And what that dye does is it senses oxygen, the depletion of oxygen, right? And then you put your TB bacteria in there, and if they start growing, what do the bacteria need when they grow? Oxygen, right? So they use up the oxygen, and as the oxygen gets used up, this little dye gets turned on, right? So suddenly the tube fluoresces, and you can see, hey, this person has TB. But because, your mouth can have other bacteria too, you can have contamination and other organisms can grow up, giving you a false diagnosis. So in most cases, when this thing changes color, we speciate it and we say, okay, this is TB or it's some sort of contaminant. And then the final diagnostic is what we call molecular diagnostics. This is how COVID is diagnosed, right? So has anyone heard the term molecular diagnostic? No? Have you? Okay, so a molecular diagnostic, for those of you who are not familiar, detects a molecule, a piece of the infectious agent, right? So culture detects the whole bacteria. That's why I showed it to you, right? The whole bacteria has to grow, then you know it's there. A molecular diagnostic, for example, can detect for COVID, it detects the COVID genetic material, right? You've heard a PCR test. Yeah, you've heard that term, a PCR test. Basically what we do is COVID is a virus and it keeps its genetic material as RNA, right? So you take the patient's swab, it's a nasal swab, you isolate the RNA, and then we have a way of converting it to DNA. And then the DNA gets detected by PCR, right? And that's a molecular diagnostic. You're detecting a molecule, right? So we now have molecular diagnostics for TB. And the one I want to talk about is this Gene Expert. It's the most widely used test here in South Africa. Go ahead.
Is that molecule just specific to COVID?
Yes, highly specific, right? Which is good and bad. Why do you think it's bad?
I know it's good, I'm not sure why it's bad.
So let's say it's in something like the spike protein, right? And the protein now mutates. You've seen, we've had beta, we've had delta, and we've had omicron, right? And if you're unlucky that it mutates in the space where your diagnostic is working, that diagnostic analyte is going to fail, right? It's not going to work.
Because if it's just specific to COVID, then what if it appears in something else?
Yeah, so it's specific for COVID. It only detects, let's say, the COVID spike protein, right? And that's perfect, right? I mean, that's how we pick up COVID infection. But there are certain tests, for example, now when the virus is mutated to give you Omicron, the spike protein itself is mutated. Now that diagnostic test that was targeting the spike protein starts to fail. Luckily, that diagnostic test, most diagnostic tests don't only target one thing. So you'll target the spike protein, and you'll target something else, right, in COVID. And so if that comes out and the spike protein fails, you can say, hey, I have COVID here in this specimen, and it's most likely Omicron because this analyte has failed, right? So having these molecular diagnostic tests is incredibly powerful for you to be able to diagnose people and monitor how the pandemic is evolving, right? And so the way they work, I'll give you the scenario for TB, and it's very similar for COVID if you just account for the RNA DNA difference. Right? So we have a sputum specimen, we have something that inactivates it because we don't want infectious stuff in the lab as much as possible. And then there's this little cartridge, right? You put this inactivated mixture into the cartridge, and that goes into a machine. And effectively, what happens in the machine? So here's an electron micrograph of TB bacteria, and there's a specific piece of their DNA, right? So we call that the rifampicin resistance determining region. It's a small 81 base pair region, and you see those different colors, right? You have these different probes, different color probes. They all bind to that region. And if all 5 of them light up, we say, hey, this is TB bacteria. And that's basically how many molecular diagnostic tests that detect DNA, that's how they work. Okay, and so you'll see these amplification curves. So if any of you have had a COVID test done and they tell you what is your viral load, the viral load gets determined by this amplification curve. You read it off. How many viral particles you get, you can read it off this curve. So how do we use, so the ability to have these types of molecular diagnostic stuff gives you electronic data, right? As opposed to looking down a microscope and say, hey, I see a lot of bacteria, this person has TB, right? Now when you have molecular diagnostics that give you this type of quantitative data, if you develop algorithms that can integrate the quantitative data into electronic dashboards, you can now start to look at how pandemics are evolving in societies and in countries, right? And so here's a really nice example from a group we've had some discussions with about modeling the spread of TB using diagnostic data in a small community in Brazil, right? So Brazil is giant and they have a huge TB problem. But so effectively, if you follow here, what you see are, this is a small region in Brazil, and what you see are district boundaries, right? These are little district boundaries. And if you imagine there are about 400 clinics in this area, and what the study did was they took all the gene expert data, and they used the clinic's GIS location. So they said, if there's a clinic here at UJ, whatever its GIS location is, and they take how many positive gene expert results they have, and they integrate that into a geospatial map. And suddenly what you see are transmission hotspots. Can you see that, guys? You see a whole region where there's a lot of gray, which means there's a lot of positive gene expert tests. And these regions which are doing gene expert tests also, and the algorithm accounts for testing volumes, right? So it's not that these areas are doing fewer tests. They may be doing fewer tests, but it's accounted for. And you can see 2 local hotspots of TB transmission emerging in this community using this approach of developing an algorithm to integrate electronic diagnostic data, right? And so this is quite powerful. In fact, this is the way we study the spread of COVID in South Africa and the way most countries are doing it, because you have this electronic data available in the public healthcare system, and you develop ways and electronic dashboards of being able to look at it at a provincial level, at a district level, and at a country level, right? So just to summarize that bit of our lecture, we know that now TB can be diagnosed using multiple methods. But these molecular methods, such as GeneXpert, give you quantitative data that then allow you to develop algorithms to map pandemics and how they evolve, right? And those geospatial models can be used to study things like transmission hotspots. But why is this important? Why do you think this is important? Right, and so I've got it there, right? So it helps you deploy public health resources. If you as government now, right? Or you look at your country and you say, hang on, I've got all these hotspots of TB. Do they have enough resources? Do they have enough drugs? Are there enough community healthcare workers to go out and make sure that everyone's getting treatment? It allows you in a smarter way, as opposed to saying, okay, I'm going to do it by the number of people in each province. I'm going to allocate resources that way. It allows you to be much more smarter about getting help to those people who need it the most, and as opposed to having resources laying around where you want to need it. Okay, and then the last two I'm going to take together, something about the bacterium and how it responds to treatment. Go ahead.
Yeah. I actually just the previous comment you made about the hotspots. Does that actually get implemented? Like, to what extent is our government aware of hotspots for TB, for whichever disease? And are they allocating resources intelligently like that? Or is this just something that we should be doing?
So yes and no. We could be better about doing it. So we have you've all heard of the NICD, the National Institute of Communicable Diseases, right? So we have what we call NICD sentinel sites around the country that report daily to the NICD.gov database as to how many gene experts or whatever it is, different molecular diagnostics, how many infections they've seen. And the sentinel sites report on everything, not just COVID, Group B, strep, RSV, pneumonia, everything. They report on everything. And then that data is fed in real time to the government. So there is the government has an approach of trying to get resources to where it's needed the most, right? But where it falls apart is at just district and province level. You know, so in many cases, like, you know, there are provinces that are so poorly equipped, there aren't enough ambulances. Like Eastern Cape, the health system in Eastern Cape is in pretty bad shape. Nelson Mandela Bay, that whole space, like the health system, even though they had really bad first and second COVID waves, and we could see it coming. You know, the health system itself is so crippled that that systemic issue overshadows any nuance that you try to build into the public healthcare system, right? But lots of countries, India is a very good example. Lots of countries have done this very successfully. The other thing that you know, so this is active, where you're looking at people who present in the clinic who are sick. The other thing you can do is also survey the environment, right? And so by surveying the environment, you can do, I don't know if anyone here has heard of the COVID wastewater program. You heard about that? So it turns out you get COVID in wastewater, right? Those people who are infected.
Australia, they have.
Yeah, in Australia they have a fantastic program. You know, you get secreted from your body and you find it in wastewater. But that wastewater actually tells you, because it tells you about everyone who's infected, right? Not just the people who were symptomatic or who were concerned or who came to the clinic, right? That tells you about schools, it tells you about mines, it tells you about everyone, right? It tells you about everyone who's infected. And you can actually model these, you can see the waves of the pandemic and you can model the before and the after by just monitoring the wastewater. And so we actually, the South African Medical Research Council has a program, and they've developed this really cool way of doing in real time this monitoring of wastewater. But I think we as biologists, we can come up with these neat tricks, right? But where we fall short is the integration of this into large data and algorithms and, you know, the computational biology and mathematical modeling. I think that, especially here in South Africa, that's still in its infancy. Often I have to call on international collaborators to help with that. And that's really where the skill that sits in the room and online is quite important. Yeah, go ahead.
So since you just showed us a model of like, since you just showed us a model of like TB and how you can forecast and all of that, how is it that we're still struggling then with taking, well, since you said that now during COVID, there was less tests done and all of that, but we know what the forecast is, right? So how are we still not working or using that in order to provide it to our community?
I think it's completely delinked. And it's a $1 million question. We have the tools. We know where the disease is, yet the disease still festers. I think that you know, the health system at community level, especially in Brazil, really needs to be strengthened. And here in South Africa too. Like, we know these communities are battling this. So TB is much more complicated than COVID. It's a long term, you know, and people can be shedding bacteria for a long time before they get sick. So they infect a lot of people before they actually get sick. So this idea of trying to find people very early, interrupting transmission is incredibly important. And you're right, because you know, if you're in a hotspot, there's a lot of transmission going on, right? So what you should do is target a transmission blocking strategy to that area. A good example would be we give everyone preventative therapy. We give everyone a drug that makes sure they never get TB, even if they're exposed, right? And instead of, in South Africa, you'd have to put something other than drinking water, because so many of us are exposed, right? But if you use this, you target the community, and there's actually 2 studies going on that look at this stuff. So we're trying to gather the empirical data to show that geospatial mapping of hotspots allows you to better deploy public health resources, and you get a better result from those communities than a community that just gets a standard of care. Yeah.
Maybe after you talk this question in the chat.
Okay, will you remind me? Okay, cool. Okay, guys, so the final components, I'm going to take these two together, the bacterium and treatment, right? And so I mentioned TB is caused by a bug. So I thought I'll show you the tree of life for bacterial organisms, right? So the tree of life for bacteria is divided into sort of these three main branches, what we call the archaea, bacteria that live in geothermal vents, very strange organisms. The eubacteria, so things like fungi, and then the actinomycetes. And so if you walk along this red tree of life, you walk along this road and you branch out here, you get these organisms called the actinomycetes. And this sort of red branch of life, where you find actinomycetes has several interesting organisms, rhodococci, corynebacteria, those of you that take vitamin supplements, most of them are produced in corynebacteria. And then you find this group of organisms called the mycobacteria. And so mycobacterium tuberculosis and other mycobacteria cause many clinical infectious diseases. Things like leprosy, I'm sure you've heard of leprosy, is caused by a mycobacterium. Burundii ulcer is caused by a mycobacterium, but TB is caused by Mycobacterium tuberculosis. And so it's this rod-shaped organism, and we will come back to this when we talk about integrated image data, right? And so when you sick with TB, right, you are actually put on to 6 months of treatment. Did you know that, right? Tuberculosis treatment is quite long-term. And we're going to talk about this now. This is really the primary focus of the problem I want to present to you. Right, so if those of you who've had a throat infection, or you take antibiotics for 5 days and you die, right? But TB, you give people 4 drugs for 2 months. So you give them rifampicin, isoniazid, pyrazinamide, ethambutol, you give them this cocktail for 2 months. And then for a further 4 months, you give them just rifampicin and isoniazid, right? And after 6 months, most people are cured, right? Now, why do we have to give people back? Why do you have to give people drugs for 6 months? What is it about TB bacteria that necessitates that you've got to treat these organisms for so long, right? And so that brings me to this idea of persistence and heterogeneity. Who's had antibiotics here? Right? And what were you told when you were given the antibiotics?
Finish the course.
Finish the course, right? And so we finish the course because bacteria have this, they display this phenomenon called persistence, right? And that persistence is really related to the fact that bacterial populations, you have millions of them infected, right? They're incredibly heterogeneous at a single cell level, right? And so I want to illustrate this using a well-known soccer icon. I'm sure everyone knows who Pele is, right? You guys all know the soccer player? Pele?
Yeah.
Right? So let's imagine that we don't have one Pele. We actually have 4 of them, right? They were, and I'm sure this would have been a wonderful world to live in if you're a soccer fan, right? So we had 4 Peles. They're all genetically identical to each other, right? They were all born from the same mom, exactly genetically identical. However, each of these Peles had a different life experience, right? This one got to play soccer, very happy. This one was bullied when he was in school. He had a very traumatic childhood. This one was very studious. He hung out with all the very intelligent kids. And this one was just a complete stress ball, right? Now, you can imagine that these different life experiences, they imprint on the different Peles differently. So whilst they're genetically identical, their lived experience creates different responses, right? And so when you apply a stress now to all of these Peles, like you tell them, go and win the FIFA World Cup, each of the Peles will respond slightly differently, right? One will be very happy, one will be very angry, depending on what their life experience is and how they've lived, right? Thankfully for Brazil, they had this one because they won the World Cup 3 times when Pele was playing, right? So the idea of heterogeneity and the application of stress, okay? So now I'm going to take that and I want to extend that to bacteria, right? So I'm going to show you a movie, and the movie has a TB bacterium, right? You see it there, we're starting off with it, it's green, right? And the TB bacterium carries a protein that is bright green. If you go to these pet stores and you see these corals, very bright colors. It's a gene from one of those corals. We put it into the bacteria, and now the bacteria turn green. So when the bacteria are green, they're alive. And when the bacteria are not green, they're dead, right? And what we're going to do is we're going to grow this TB bacterium, and then we're going to treat it, right? And then we're going to take the treatment away, and I want you to see what happens, right? So here's it growing with no treatment, right? And you see 1 becomes 2, 2 becomes 4, and the bugs grow. We now give it treatment, right? So this is you taking your drugs. See what happens? The first thing that happens is they stop growing. Yeah? Then a whole bunch of them die. This is you taking your drugs now for the first 2 days or something, right? Then what happens is, do you see that? Some of them hang around. You see that? Look at that. They haven't died. The first lot died very quickly. Oh, sorry about that. Let's try that again. Right, so then they're growing. And then you start taking your drugs, they stop growing, and then some of them die. Right? So within the first 2 or 3 days of treatment, a lot of the bacteria die, right? And then you see some hang around. And you see that they don't die, they hang around, and some actually grow in the presence of the drug. There's one fat one down the bottom. It's like growing slowly, and it's dying slowly, but it still needs the drug, right? It's not dying quickly, right? And eventually you see it will die. And then that one on the top corner is also growing. Do you see that? It's growing slowly. So even though you're giving the bacteria drugs, they're still growing, right? And then eventually this fat one in the middle dies, right? So, and then you see another one just popped over there, right? So this is the idea of persistence. You see the bacteria persist, right? They don't die when you give them the first round of treatment, and you've got to continuously treat, right? And you will continuously treat, and there are 2 populations, one that actually dies very quickly, and one that appears to be hanging around. Now you take away the drugs. Let's say you stop taking your drugs. Look what happens. Right? And they're not genetically resistant, because when you start giving them drugs again, they start dying, right? So you see this is the idea of persistence, right? These bacteria and organisms that don't die when you give them 1 round of treatment or treatment for a few days, they actually need to be treated for a very long time. Now, this is the holy grail in TB, right? If we can find a way of taking the 6 months of treatment and shortening it, we make people's lives better, we improve the quality of healthcare delivery, and we can actually make a difference in society, right? But in order for us to be able to shorten treatment, we have to make sure we kill everything. We don't want to shorten treatment, and then people start getting sick again after you withdraw treatment, right? So I want to bring you to this concept now of culturability. So go ahead.
Would the longevity of that decrease if it were easily detectable? Or like, since you said it takes a while before people actually get sick, so would that decrease if maybe like you can detect it as soon as it's like a small molecule or something?
Okay, so that's a fantastic question. You've preempted me. Okay, so that's what I'm going to tell you about. How do we detect these bacteria? And can we detect them quickly? So normally, what you'd like to do, if you presented with TB in the clinic, right, and you gave me a sputum, I would like to look down the microscope and say, you have these bacteria that are going to need treatment for longer, right? And so you need longer treatments, right? But you come in and I look down the microscope and I say, you don't have any of these. 2 weeks of treatment is enough for you, right? So I don't over-treat him and I don't under-treat you, right? Right now I have 1 blood instrument that gives everyone the same thing, right? So can we detect these bacteria early on, right? And actually guide treatment in a precision medicine way, right? So how do we differentiate the ones that grow fast and die quickly and the ones that don't die in the presence of antibiotics, right? That's the million-dollar question. So I've taken a frame of that movie, and I want to give you a sense of how these organisms now behave when we treat them in the lab, right? So if you take the ones that died, right? You see the ones that died? If you take the ones that died and you try and grow them on either solid media or on liquid media, they actually grow, right? You put them and you get these little colonies in solid media and they grow on liquid media. The ones that die appear to grow quickly, right? When we try to look at them in the lab. But these ones that hang about, right? When you put them on solid or liquid media, they actually don't grow on solid media. The ones that need long treatment don't want to grow, right? And in liquid media, they're lazy. They don't want to grow. You have to give them something to get a kickstart, right? So almost in a way, it's like we give them Red Bull, right? And so what does Red Bull do, guys? Red Bull gives you wings, right? So if you give them a bit of Red Bull, then they grow, right? And so these bacteria that require long-term treatment, they actually have this thing that we call differential culturability, or DCTB. They're persistent, right? They don't have equivalent culturability, so they don't grow as well on solid and liquid. They only grow in liquid media. and they grow when you give them something to grow, right? So can we detect these DCTB bacteria directly in sputum, and can we develop a quick mathematical algorithm that if we feed an image into that algorithm, it will tell you there are DCTB there, and this person needs treatment for longer, or a very few DCTB, give treatment for 2 months, monitor clinically, and leave the person alone there, right? So how do we detect these DCTB, right? So from here, you know they only grow in liquid media, right? So how do we quantify things in liquid when we can't plate it out and count 1, 1, 1, 1, 1, 1, 1, right? And so the way we do it is we use this thing called the most probable number assay, right? So it's based on the Poisson distribution. So imagine you had a sputum sample, and there were 100 bacteria there. Now, without plating it out, how do you know there are 100 bacteria there? So you do a dilution, right? So if you did a 1 in 10 dilution, right, and you made a series of dilutions like that, so you'd start off with, in the first dilution, you take 100, you dilute it 10 times, there's 10 there, then you dilute it again, and there's nothing. Between 1 and 0 and nothing, right? And so if you did that dilution perfectly and you gave a bit of Red Bull, those 2 tubes will grow up. First 2 will grow up. And if the first 2 grow up, you'd say, you know what, there's about 100 organisms in here, anywhere between 100 and 900 around. You can get a reasonable confidence limit around it, right? But scientists are not perfect. Those of us that work in the lab are not. Our pipeetting is not perfect. So usually what we do is we say there's 1 to 100 CFUs there, right? But if that third one flagged positive, just because we didn't pipette well, right? You erroneously say there's 1,000. So in order to get away from that and get around that problem, we just set up a lot of things. You do this in multiples, right? And you grow these things up, and if you have enough that give you the first 2 tubes positive, you say, actually, I have between 1 to 100 CFUs in there, right? I can work out how many DCTB there are, right? Now, this is a cumbersome way of doing it, right? We want a better way of doing it. And so I want to give you a sense of how I set this up in the lab, and then how we generate imaging data from it, right? So in the lab, you want some Red Bull, right? You want something that allows bacteria to grow. Now, what's a good signal? If you were a bug and you didn't want to grow, what's a good signal to grow? Sometimes people say food, right? If there's food around, you grow, right? But another good signal, like humans, bacteria are social creatures, right? So they like to follow the norm. So if other bacteria are growing, then those that are not growing want to grow, right? So what we do is we take a culture of bacteria in the lab, we grow them up, right? And when they're growing, they secrete these molecules and say, everybody grow, right? And then we take out all those bacteria, and we have what we call a culture filtrate that has no bacteria, but all those molecules, all the signals the bacteria are saying, everybody grow. And then we take that, and that's kind of our Red Bull. It's what's going to get the bacteria to start growing, right? We take that, and we, as a control, we just take regular lab media. We apply it to this 48-well plate, and then we take a sputum sample, we do that 10-fold dilution, and you add this juice, and suddenly all these DCTB in sputum that don't want to grow, they start to grow, right? And you can quantify them. They're coming out, and they allow you to quantify them, at least with a reasonable degree of confidence, right? And so you then take this incidence of positive and negative growth. You know, we've got this well that's shown growth and no growth. You plug it into the Poisson, and that allows you to determine an actual number, what we call the most probable number, right? You divide that by the number of bacteria you can count on a plate, and you get what we call the DCTB, right? So I hope this makes sense to you, but it's a very cumbersome way of doing it, right? So I want to show you a possible better way. But to give you a sense of what this looks like in the lab, this is a patient we had from one of our clinics in Soweto. And you can see that, you know, under the conditions where we didn't give any Red Bull, nothing grew. And I hope you can see this. Can you see that growth? That white stuff? Those are TB bacteria that are growing, right? And you can start to now quantify this, right? So you go through, you pick out the positive and negative growth. This gives us 1,000, most probable number. There were no colonies here. This person only had DCTB. And so we say there were 3 log DCTB, and they required culture filtrate to grow, right? And you can put that as a little red bar, and then there were no bacteria that grew under normal conditions. And so if you look at this is about 40-odd patients we looked at in about 6 clinics in Soweto. And you can see most of them, if not all. So with the exception of 3 or so, we had 14 one clinic in Soweto and 14 another clinic in Matlosana. You see that I'm showing you the first set of data that most of them have these difficult to culture bacteria. And so when we put them onto treatment, chances are they're not going to respond quickly. So we need to monitor them and give them treatment long enough so that they actually get cured, right? So I want to build this model for you. Sputum has a lot of bacteria that don't grow. And in order for you to grow them, you need to add this red ball, right? So when you now follow these people, to answer your question, right? When you follow these people longitudinally, right? And you look at the red ball population and something that we can conventionally count on plates, you see two like sort of responses. You see people who the red ball population never clears. You see here, this is just normal growing bacteria. So these are the bacteria in the movie you saw that died quickly. In purple, they shown here. And you have them clearing in both of these groups, but you see this red ball population here doesn't clear and eventually starts to clear slowly. And here it actually accumulates, right? So you have people who, even though you put them onto treatment, it looks like they're getting worse, right? So these red lines represent those organisms at a single cell level that weren't died. And so we want to quantify them early on, and we want to be able to direct treatment in a more careful way, right? So if you now map onto I told you we were working in Soweto. If you take a map of Soweto and you say, okay, well, how many different types of TB strains can I find? So like COVID, you have alpha, beta, delta, omicron. With TB, you have lots of different strains. Right? And I've just shown you 9 there. Right? In South Africa, these are the prevalent ones. And so we thought, well, is it something about a particular strain of TB? Omicron doesn't make people sick so much, right? Is it something about a particular strain of TB that generates these DCTB that makes it difficult to detect them? And so what we did is we went through, spatially pulled out all these different clinics, and we said, well, how much DCTB can we find by strain type? Right? And so those are all the clinics, that's where we worked, and those are the strain types we found. And then we quantified the amount of DCTB you could find. And so you see, actually, there are some strains like this Beijing strain, which in South Africa is associated with severe disease. It produces a lot of DCTB when you compare it to something like the land strain. So now we're getting closer to something we can operationalize. We don't want to have all these hundreds of tubes and dilutions and Red Bull going on in the clinics. We want something we can look down the microscope and say, this person is not going to get better, right? So it looks like if they have a certain type of strain, we can actually tell that certain type of strain is not going to respond well to treatment. But now by microscopy, how do we distinguish one strain from another, right? How do we do that? That becomes tricky. And so that can I carry on? Really?
Yes, Prof. I just want to remind everyone, please stay muted. So there's no interference here because we're in a room with which is very sensitive to audio. Thank you, everyone. Prof, you can proceed.
Okay, thank you. So the type of strain can influence your ability to find DCTB, right? But can you use this information to develop better diagnostics? And so I'm going to bring you to the last part of the lecture where in order for me to explain to you how we do this, I have to tell you how these TB bacteria divide. Okay, so I know it's an overload of biology. This is my last bit, but it's really important for the final punchline, right? So imagine these bacteria, right? These rod-shaped bacteria, right? And in order for them to divide, they need to expand, right? And they need to expand and they need to incorporate new bits of cell wall. So I've got a little cartoon. So there's the bug, right? And there's its cell wall shown in green, right? And so the way TB bacteria grow is they put new cell wall at the pole, shown by the little light green, right? And so you see that new cell wall occurs just at that subpolar region of the pole. And as the bacteria grow, they incorporate new cell wall, then they push out, right? Then eventually for cell division to occur, right, you have these gradients of cell wall, right? The new cell wall at the end, because the bugs have put it on there, and then the very old cell wall in the center. Make sense so far, guys? Yeah? But we still have one bug, right? You want the bug to divide. So what the bug does then is it synthesizes a division plane, right? That separates the mother cell into two daughter cells, right? And then you have enzymes that break down that bridge and they eventually allow these bacteria to divide, okay? So very basic. You have extension of the new cell wall, which allows the cell to push out, right? You have a division bridge being synthesized, and then you have that being broken down. But why is this important? It's important because when you look at now in the daughter cells, when you look at the new cell wall versus the old cell wall, right? You actually have this new cell wall at the pole. And that's important because we have a stain that allows us to detect the new cell wall, right? And so you apply that stain, and this is an actual TB bug. And can you see that, guys? Can you see that image? Where you have that new cell wall and cell pole, and there's a gradient of it going towards the center, right? So that's how we would expect a TB bacterium to incorporate new cell wall when it's growing. So far, so good, everyone? Yeah? Okay, so some questions now arise with our DCTB problem, right? If DCTB do not grow, do they take up these stains, right? Can we use these stains to detect them? If they do stain, can they stain differently? To answer your question, can you find them quickly, right? Are there any strain-specific effects? Can we pick out, you know, like with COVID, can you pick out different strains just by the basis of the way they stain? And then can this imaging be used to develop an algorithm that would serve as the basis for smarter diagnostics, right? And so for this, what we did was we actually developed a model in the lab that allows us to create DCTB, because we don't want to be using stuff from people all the time, right? So we take bacteria, we culture them, and what we do is we wash them in saline. So you take away all the nutrients, everything, right? And you starve the organisms. And when you starve them, they actually develop DCTB that you can resuscitate, right? And so as an example of this, what we did is this is our lab stray, and we pushed it through this model and we allowed it to grow out. And I hope that you can see, I'm going to quantify for you here on this graph, this idea of the hot poles and the side wall. The pole must be hot, right? Because it's got new cell wall. And I hope you can see that here, right? But the idea is to develop computational ways to look at it better, right? But like, if you look over here, do you see that, guys? Do you see those hot poles? Yeah? See that? The hot poles and the division plane? Right, so this is our lab stray. It incorporates these hot poles, right? And if you, this is the kids in the lab, and we do this manually. You go through the image and you go through the bugs one by one, and you say, okay, this is the number that have hot poles. This is the number that have hot side walls, right? Now, when you take, remember I said we had a lot of Beijing in Soweto. Remember that, guys? We had a lot of that Beijing strain, right? So when we took that Beijing strain for one of the patients we found in Soweto, and we pushed it through this model, it looks very different. Do you see that? Doesn't seem to have hot poles. It seems to be, it's got punctate distribution of the stain along the side wall. Look at that, there's all these dots around here. Then you have here, the poles are completely cold and the side walls completely hot. Do you see that? Right, so when we quantify this in a very rudimentary way, just manually, you can see that this Beijing looks very different, right? It doesn't have hot poles. It's got a hot side wall, right? And these people, you know, actually didn't respond well to treatment because we followed them up, right? And then there was that other strain I told you that made very little DCTB. That was the lamb strain. We find this a lot in people who have HIV-associated TB. And so, for example, in a place like Matlosana, where there are a lot of the mines, Matlosana is home to some of the world's deepest gold mines. There's a lot of TB there. And you see the lamb strain now looks also very different in that it doesn't have hot poles or hot sidewalls. It seems to just be hot everywhere, right? Look at that, it's hot everywhere, right? So this is actually the basis, I believe, of trying to develop algorithms that can allow us to develop smarter diagnostics, right? So if we stain these organisms, you walk into the clinic, and we stain these organisms with a stain. So we're working with a group in San Francisco that have 3D printed us a little microscope that can fit in your pockets, right? And you can take this to any clinic, anywhere. It does not rely on heavy resources. You take the image and you develop an algorithm that very quickly says, hey, this stain is being incorporated this way. Most likely this is a Beijing strain, and it's not going to respond well to treatment because it makes a lot of DCTB. Right, we've got all the biological evidence to support it. I think where we now need help is how do we develop computational algorithms that can do this quickly and with a reasonable degree of accuracy. Okay, and so that brings me to the end. I just want to summarize this bit. Persisters, remember, take your drugs, guys. Persisters are drug tolerant. You need to be able to finish your treatment, but they're hard to culture. So we can't tell from a context of chronic diseases like TB, we can't tell who's going to respond quickly and who's going to respond poorly. And it really comes down to quality of life, right? This is you know, if you ever knew anyone with TB and you knew the pills they have to take every day, and if they have to take them unnecessarily, right, that's quite sad. It's really not a necessary thing. If we can improve the way we deliver diagnostics and deliver intervention, I think we can make a big difference. And then we can possibly use the way these cells stain to identify them quickly and tailor therapy. And perhaps that machine learning approach can be used here that I believe is something you guys are going to be talking about. right? And then just as a final thing, I thought, I'm sure, I was told that postgraduate students here, people who work across a lot of disciplines. And I thought, well, I'm going to just give you as a parting shot my own view of the future of healthcare, right? And where I think a lot of computational biology and digital health fits in, right? So this idea of the patient and the doctor, which is the traditional approach of how we deliver medicine, right? And COVID has fundamentally changed that, but that was changing already, right? And it's changing because of the following things, right? We have the advent of these wearable devices, right? I'm sure the wearable you're familiar with is your Fitbit or something, right? But the technology has gone way beyond that. This is a new thing that's come out in China. You can actually, and maybe it's relevant for South Africa with ESCOM, but you use your body energy to charge the Fitbit. You do not need electricity, right? It's got these serpentine coils that harness the energy, the heat energy from your body, and it turns that into an electric charge that allows you to charge your wearables, right? The idea of mobile health apps. So there's an app that we were working with a group at UW, right? So it's actually a cough acoustic app. So your phone is an amazing device, right? I don't have mine here. Do you have your phone? You know, so if you look at this thing, right, it's actually an amazing device to deliver healthcare, right? You have a heat-sensitive surface, you have several cameras, and you have a very, very sensitive voice recording device, right? So we have a group at UW that have developed a phone spirometer. So you blow on the phone, and it tells you about your lung function, right? If you put your finger over the camera and the flash, it tells you a red blood cell count, right? But the voice recorder can actually record your voice and the acoustic nature, thank you, and the acoustic nature of your cough. So we have an app that it runs in the background, right? And so it's on your phone, it's running all the time, like WhatsApp, and it's recording your cough. And after a day or two, it says, hang on, Bhavesh, you know, it's not just the cough, it's the frequency of cough and the acoustic nature of the cough. So is the cough coming from a cavity, or is it just coming from your upper throat? It will say, hang on, Bhavesh, you know you've been coughing with a frequency of more than 40 coughs per hour for the last 3 days. And the cough sounds like it might be a TB cough. And it geospatially links you to the nearest TB clinic, and it tells that clinic you're coming. And if you don't come in 2 days, that clinic now has your mobile number, they phone you, right? And so healthcare is being revolutionized, I think, by the advent of digital health approaches, right? And then there's this idea of telemedicine, right? And we tried this, I think, woefully, inadequately in South Africa. We tried distance. Can you paint the picture of a patient from a distance when COVID came, right? You tell us your symptoms and we'll tell you whether you had COVID. It didn't work well. But that idea is really taking hold, I think, for diseases that are easier to manage using that approach, right? And then digital adherence and support technologies, right? So one of the things we have was TB. So if you have HIV and TB together, right? You're taking ART and you're taking TB medication, right? And it's really very challenging for these people. But what some of the ARTs do is they reduce the bioavailability of TB medicines, right? So you want to be able to make sure people are taking the drugs, right? But you also want to know that there's enough in the blood, that the drugs are having a therapeutic effect. So now we have these things called digital adherence technology. So you can put in the phone how many times you take your drugs, and the clinic can monitor it. But also, you know, there's this thing called a MERM box. Your phone is linked to it. Every time you open the lid to take out the pill, it records it. So your doctor knows, but you can lie. You can open and close it and not take it, right? But your doctor knows, hang on, this person is taking their pills. You can even, there's a little strip test. You urinate on it and it tells you, and it's got a color matrix and it tells you how much drug there is in the system. You take a photo of it and the phone integrates the image and it tells you, you know, you're getting enough drug in your system, right? And then the idea of electronic health records, right? So there's a lot of the COVID geospatial mapping is an example of electronic health records, right? So I want you to keep this in mind when you think of how you apply your skill, right? Healthcare delivery is fundamentally changing. Sorry, go ahead.
Yeah.
Okay, one more slide, right? There we go. So some things for you to think about, right? I mean, there's a rapid increase in these technologies, but the pace of the local innovation is slow, right? It's somewhat out of step in terms of what the world is doing with digital health. And whilst recognizing how many times have you not doing the 4IR and doing the 5IR, but the local funding to support these things is somewhat out of step, right? And the reason I came today is because I think the coordination between disciplines is poor. We need to be talking to each other more. Clinical evaluation is incredibly costly. So if we can do it computationally, that really gives a leapfrog to allow you to implement some of these interventions. But also the legislation is evolving quite slowly. So I tried to get the powers that be convinced about this phone app, but the legislation around data privacy, around how you access this type of data, it's not in step with being able to implement this thing. So to answer your question about, we know the information, but can we implement it? So for example, if we can map a geospatial hotspot using the data people give us from their phone apps, if they're diagnosing at home, right, we actually need their permission to use that data to deploy healthcare resources. That's how the legislation is written currently. So somehow these two things need to start speaking to each other, the legislation and the public healthcare need. And so the challenge is to provide an integrated cross-disciplinary platform that allows you to incubate, develop, and deploy this new era of digital health interventions. Thank you very much.
Well, Prof, I promise one of the questions here. So between the MGIT test and Gene Expert, which is more reliable? If one test is positive for TB and the other isn't, which one do you believe? More generally, is a molecular test better than a culture test or vice versa?
Oh, that's like a fantastic question. Even though we have these molecular diagnostics, we still say culture is the gold standard. Right, so we can have people who are gene expert negative and they culture positive, but we also have people who are gene expert positive but culture negative. So culture is the gold standard. That's what we use as the most reliable diagnostic.
Any more questions from the audience? Yes, Prof. Panasha.
Hi, thanks for very good talk. In some sense, showing the other side of the story from mathematics. Now I have two questions, not mathematical questions essentially. One is, in some sense, all anti-COVID measures are similar to anti-TB measures. Have you witnessed, decrease in the number of TBs because of use of masks, social distancing, and something like that, or not? Or somehow it doesn't work like that?
Yeah, it doesn't work like that. So the problem is that, so we can only say that there's a decrease in TB if we haven't decreased the amount of testing. So if I did 100 tests and I found 70 TBs, and then now in 2021 I do 100 tests and I find 30 TBs, I can say there's a decrease in the number of tests. But what we did was we were doing 100 tests, we found 70 TBs, and now we're doing 30 tests. And so it's hard for us, if you're not catching the same number of people, it's hard. So TB incidence has dropped. But we don't believe it's dropped because it's getting better, it's disappearing.
Yeah. I mean, for me, maybe the positive effect of COVID will be that people start washing hands, more regularly than before, and things like that.
And that has happened. Yeah. I mean, we effectively eliminated flu, right? Like, so last year and this year, last year and the previous year, the flu pandemic that would happen in our winter season has significantly diminished. So that has happened.
In Europe last year, the number of excess deaths dropped in the first three months of COVID because people stopped using cars, and then suddenly the air became much cleaner. So I mean, there are positives of COVID. And the other question, comment, you in the last couple of slides you showed us bright future of medicine, but this is essentially what is about all protests against COVID restrictions, because suddenly when I look at your vision, it's like Minority Report, like being under total surveillance all the time. I cannot cough without, local police commissioner and knowing about this. So I mean, it will be big backlash.
I was thinking of what you were saying about you can't, even if you can use people's data to get the hotspots, you then need their permission to deploy. And I started laughing because I thought to myself, so basically I'm not allowed to die by my own hand if I choose to. I'm going to force your healthcare on me against my will. And it becomes a debate, like to what extent can I say, no, I don't want healthcare, even if I have TB, just leave me alone, versus like you're saying, I mean, there are so many instances where people are falling through the gaps because we just don't know. But at the same time, there's that privacy where you are trying to enforce something that it's not necessarily
I've seen your hand. Yeah. So I think that there's two sides to this, right?
Yeah.
So the idea of digital healthcare and delivering digital healthcare, the primary purpose is to put the power in your hands, right? So you have and this is we have to move to society where people feel like they're in charge of their own healthcare, right? And that you're not putting that in the hands of the health system, but you're putting that in the hands of the government, right? And so the idea of having like a phone app, for example, you have, so when you go to the doctor looks at you in that 20 minutes or so and paints a picture of you in that 20 minutes, right? Your phone app will paint a much more accurate picture of you because it's giving you a picture of the time, right? Of course, it's still your right whether you want to choose to share that information or not. The idea is the power is in your own hands. Right?
And then you use Pegasus of these surveillance systems, developed by Israelis, can easily break into any security.
Yeah, data security is always an issue. Data security, and that's why the legislation is so strong, right? Because protection of personal information is really important. But it shouldn't be a deterrent to move away from something that can make society better, right? And then the idea of you have a community that's in dramatic need of care, right? One of the biggest problems in South Africa, in Africa, and in the developing world is we are, for some reason, unable to get help to the people who need help, right? And that divide of the people who have and don't have, the divide of the people who don't have help and the people who have access to stockpiles of vaccines, right? That divide has just become worse over the last 1.5 years, right? And so you need low-cost ways to be able to put the power into people's hands, right? And I firmly believe this is the way of the future, but the legislation is important, right? And why I say it's out of step, because it's not evolving with the need, right? So very soon the need will change, or the legislation will become so obstructive that there'll be no incentive to continue to develop these things. But the primary idea of digital health and precision medicine is two things: is to put the power back in the hands of the patient, but also to remove the patient from being just a number in the healthcare system. This drug dose is not going to work for you, but just because the formulation is that, we're giving it to you, right? The idea of moving to precision medicine, right? I firmly believe that's the way of the future. Sorry, go ahead. Shane, you've been.
It's basically based on what you're saying. So since you said like TB, for example, is most prevalent in the poor communities, like Eastern, said TB is most prevalent in the poor. So then how are you, with this implementation, how are you taking into account the poorer communities? If, for example, how with the foam detection thing, is it going to require a smartphone?
So yes. Yes, but did you know? So I actually checked this with the CASA last year when I was trying to develop the COVID Act. The penetration of smartphones in South Africa is 76%.
And in Eastern Cape specifically?
I don't know in Eastern Cape specifically. But that's a very important question. But I think also we must move away from this notion that sufficient technology is not there, right? The big question is, and we did a small survey, right? That would people use their data for this? And they wouldn't, right? So they wouldn't use the data for something that they don't deem to be immediately useful for them. So even though they have the phone, they're not going to use the data. So the issue, I think the biggest barrier is to make data free, right? And that will, even wherever you don't have penetrance of smartphones, that will push the penetrance of smartphones. But the availability to deploy this technology, the availability of hardware is definitely there. Yeah. So there was a hand here? Yes.
Yeah, okay. Thanks for the presentation. I want to know if you have some data sets. If I want to work on some of those things, you have some available data sets one can use?
For the imaging data? Yeah, so we had a lab flood. We couldn't locate it, but we found it. We do have images on a hard drive that you can work with.
Maybe I can also help you answer the question, because Professor Hari is the director of DSET.
Maybe we can do this by coffee.
Yeah, so
Yes, this is actually
we hope the
more
data
about-
will actually be with health data. You need to sign lots of pieces of
No,
paper.
it's just one. We have.
Oh, it's just one. Oh, okay.
So it's a confidentiality agreement. We've de-linked it from all the patients' identifiers, but it's just a one pager. That's a confidentiality agreement. We couldn't find the data. We had the top floor in our building there. You know, when Gauteng didn't have any water, they left the tap open, and then when the water came back, overnight it flooded the whole place. But we do have data. Thank you very much. Thanks.
Thank you.
For the audience in person, you can ask Prof. Kana more questions during the break. We have another speaker at 11 o'clock, Sharice, or should we 11 in 13 minutes, or do you want to? Okay, so we'll start just about 11 o'clock for the next speaker. And the people online, hope to see you in 13 or 15 minutes or so. Thank you.
Okay, so those online, please let me know if you can hear me. Anyone online can just let me know, please. They can?
Yeah. Yes. Okay, thank you.
Okay, welcome back, everyone. Our next session is going to be given by Mr. Marcel Sabming with the following title. Okay, so Marcel is a PhD student and so his specialty is machine learning. Marcel, if you can just say more or if you can start your presentation.
Hi, hello everyone. My name is Marcel Sabming. As you can see there, my supervisor of my PhD is Dr. Rodin Heyman, and my PhD is co-supervised by Dr. Vivian Sen. So today I'm going to discuss categorical and numerical data pre-processing for topological data analysis and machine learning classification. Just to mention, I'm not actually going to apply TDA on the data that we're going to pre-process for machine learning. So during the practical slide, I'm just going to actually show or demonstrate how the pre-processing works using Python. Right, as an overview, I'm going to talk about the data description. So also just to mention, the data I'm going to use is not a biological data. So it's a data that I got from a website called kaggle.com. So it's a website that offers that allow people to get into some sort of competition. They provide this free data set that you can use, and then if you clean it, apply machine learning, and then they actually grade your score. Okay, second, I also discuss handling of irrelevant features, talk about the future engineering, talk about handling of missing data for numerical data, handling of missing data for categorical data, and encoding of categorical features as well as feature selection. All right, for data description, like I said, I'm using the data from a website called Kaggle. So specifically, it's a Kaggle Titanic data set. The reference is there if you want to visit the website. And what the data set looks like, something like this. You have 12 features, you have 891 records, and you can see they contains both numerical and categorical data. And also there's some missing data. Right, so the first thing that I need to mention here is that your output is the survival of the passenger. So it's basically a simulation of the Titanic incident that happened in 1912. I'm sure everyone knows about the Titanic ship that sank. So this is just a simulated version, all right? So they give us 891 records and 19 features. And what we're going to do is to try and look at this customer feature or passenger features and try to predict if a specific passenger is going to survive the accident or not, right? So looking at the table there, you can see there's number from 0 all the way to 11. It shows actually that there are 12 features in total. The first one is just passenger ID, is just the identification of each passenger. The second one is very important, it says survive. So survive, there is a Boolean value, it's either 1 or 0. 1 means the passenger did survive the accident, 0 means the passenger actually died or sank. And then P class is the passenger class. So the three class, there's first class, the second class, and the third class. So first class obviously is for the very wealthy people, then the second class for the middle class people, and then the third class is the lower class or the poor people. And then name is just the name of the passengers. You have sex, just basically gender, is it male or female. Then age, okay, age is also given within the range of 0 to 100 in this case. Actually, they mentioned there's actually a floating value. And then the next one is the number of siblings or spouse. So for each passenger on board the ship, we also take into consideration the number of siblings or spouses that they have on board. All right, then the next one there is the number of children, parents and children. So for every passenger, we also compute the number of parents that are there. If both parents are there, just one parent, the number of children. Then it is about the ticket. So ticket, so just the passenger ticket number. Fare is how much this passenger is going to pay. Obviously, if you're a first class passenger, you're going to pay more compared to someone that's a third class passenger. And then embark is basically the port of embarkation, and there's 3 in this case. Is basically Quebec, Queenstown, and okay, the third one actually lost track of it. But the whole idea is to look at all these features, pre-process this data, apply machine learning, try and predict if a particular passenger is going to survive this accident.
Can I ask you a question? I mean, if I remember, it was close to 1,500 people who died
Yes.
on Titanic.
Yes.
So not 500. So
Yes. You're very correct. So actually, it was 1,500 passengers that died on board a ship that carried 2,200 passengers. Like I mentioned, this is a simulated version. Right? It's a small chunk of the data. All right. So I mentioned that they are missing data. Yeah, I'll just show down the next table. The next table shows how much missing data there. You see under the age category, you see that up to 177 passengers they didn't mention what their age is. And in terms of cabin number, we have up to 687 cabin numbers that were missing. And in terms of port of embarkation, we're missing 2 there. All right. And then in terms of the balancedness of the data, all right. So we want to see if this data is balanced or not. In terms of those that survive, again, this is based on the simulated data that we're using. It's 342 that accounts for 38.3%. And those that did not survive is 549, that's 61.2%. So clearly we can see that the data is clearly unbalanced, right? Now we want to handle irrelevant features. So given any data set, the first step of pre-processing, just like Prof. Hali mentioned yesterday, we said the inspection part, we need to look at the data and see if there's something really dodgy happening, like for example, an outlier and so on. You also need to look at features that are irrelevant. For example, in our case, passenger ID. So passenger ID and passenger name, clearly they can't help us. So they don't contribute in any way in the prediction of a passenger's survival, all right? So it doesn't matter what your ID is or what your name is, that doesn't help us in any way. Okay, if we look at the missing data, so there's cabin. So the cabin there, it contains 687 missing values. That's almost 77.1% of all the cabin numbers that are missing. Now the question asks itself, do we delete cabin column? All right. The answer I'm going to say based on the research that I've done for my master and my PhD is yes and no. So I would say yes if you're using a very large data set and deleting this column does not affect the number of input features that go into your machine learning. But if you use a very small data set and you compute this algorithm to actually delete all those columns that have missing values of up to 70% or more, then you might end up with just 1 or 2 features that actually goes into your machine learning. Therefore, my answer is yes or no. And then if you look at each column, so it's just 177 missing values, that's 90.87. Clearly, this column we can delete this, all right? We're going to look at the methods of dealing with missing data. And if you look at embark, they just contain 2 missing values. Again, we can't delete that column. Right, so the picture there is just showing you how many of those features are actually missing. Now, what is feature engineering? So feature engineering, we want to be able to create new features from existing features. All right, so we use information from the features that we have to generate new features. For our case, so for this specific data set I'm looking at, there's 2 possibilities. For example, I can start by saying, what if I compute the total number of missing values, which is say not a number, in each record? So I can use that as a new feature of its own. I can also have a binary feature that indicates the presence or absence of a missing value for each future instant. So you see, these are two possibilities. Again, it doesn't guarantee that this is going to improve your accuracy at the end. And the cool thing about pre-processing is that down the line, if these two extra features that are actually added doesn't contribute anyway, they get removed, all right, during the phase of future selection. All right, handling of missing data, that's for numerical. So we look at some of the popular type. The first most popular type is replace all the missing data of that specific column. Keep in mind, I'm talking of numerical data. So look at the column, for example, age. Age is numerical. So I can replace a missing age with the mean of the whole column, all right? Next, I can replace it with a 0 or just a constant value, all right? Next, we can also use the median and as well as the mode, all right? Just to mention, so median is just a middle number, the mode is just called with the highest frequency. So the age that appears the most number of times.
So if you replace with any of those, it doesn't really affect the spread of the data or anything like that. It's like, for example, the mode, that's the matter. What if the mode is like also the number that appears the most, and the highest number?
Yes. So the mode in this case is the age that appears the most number of times.
But let's say the mode is, let's say you have a spread of from 1 to 36, and the mode is 36, and that's the highest number. Will it not affect the spread of the data if you replace the mode?
Not necessarily affect the performance. It may and it may not, but keep in mind that if you give in an input to machine learning with a null value, it cannot work, right? So at the end of the day, you need to do something about it. Either you delete the entire feature or you replace it with one of these algorithms. And if yes.
Sorry, just to give you context, this is exactly where you need to decide what makes sense. So will it change it drastically? Like Marcel is saying, you can't not do anything. You have to do something. But there are a variety of options, like it's listed here. You've got to decide within your context which one makes sense and which one is just going to cause absolute havoc if you do it. So it's never just a blind. That's why, like, just like Marcel is now listing, there's always all these different kinds of options that you can go through. So yeah.
All right, and also something I need to mention is for my PhD, I actually implemented each of this method all the way from this stage right up to the end, and I choose the method that gave me the best accuracy. So from onset, you can't just say, I'll stick to mean. You have no idea if actually median might give you a better accuracy. So you can implement each of those. You just run the loop. You say replace this by mean, by median, and so on. And they have other techniques which I've seen people use. Here we replace the null values with the mean. So I look at this numerical column, I replace it with the smallest value -1 or the maximum value +1. And some people actually got very good results using this method. Now, how do we handle missing data for categorical features? It's different from numerical, or I'm going to mention it. Categorical, you're talking about text, for example, gender, male or female. So you can't say I'm going to replace it with the mean. It doesn't work, all right? So in this case, some of the most popular way of doing it is most frequent, all right? So you need to replace the textual data using the textual data, not numerical. So in this case, you can actually use the most frequent to go through the column, the specific category that appears the most number of times, you just actually use it, or you can just use a constant value again, all right? There are other ways of doing this, but I would say these are the two most popular ways of handling missing data and categorical features. Now, encoding of categorical features. Keep in mind that most machine learning algorithm does not accept text, all right? The one numbers, the one numerical input. So what you need to do is to convert all your categorical features into numerical features. So we do a process called encoding. First method is called one-hot encoding. Now, what is one-hot encoding? I'm just going to give a brief description. So we add binary values depending on the presence of or the absence of each category. And in that process, you create a new set of k-dummy variables which is equals to the number of categories. So I'm just going to explain what that means. It means that, for example, if the gender is male or female, if I encode that, I'm going to use 2 bits. All right, I'm just going to use 2 bits. I'll say 1, 0 and 0, 1. As a demonstration, they have red, green, and blue. You see, if I apply one hot encoding, because the 3 categories, I have to encode each of those using 3 bits. All right. Then there's something called dummy encoding. Now it looks similar to one-hot encoding. So it's similar to one-hot encoding, but it actually creates k-1 dummy variables for k categories. So it's always 1 less binary bit, which is really useful because you want to reduce your computational time. All right, computational complexity and computational time. And as a demonstration, again, you see the same red, green, and blue. Using dummy encoding, I just need to use 2 bits for each color. All right. So clearly, in this case, you're going to choose a dummy encoding. All right. Now, another method is called hash encoding. What if those 2 methods is not? I'm going to explain why in some case it won't work. Like specifically for my PhD, the dummy variable and the one-hot encoding, they didn't work because I was using a data set that has 230 variables and 50,000 records. So clearly, if you try to use one-hot encoding, your data set is going to multiply itself almost, you can do the math, almost 1,000 times. And we don't have that kind of processing power yet. All right. So we stick to hash encoding if the data set is very big, right? So hash encoding, how does it work? So here we can encode using hash algorithms such as the message digest, so MD2, MD4, and MD5. By default, if you're using Python and you don't specify what your hash algorithm is, it's going to use MD5. You can also choose to use the secure hash function, which is SHA-0, SHA-1, and SHA-2. Another method is called frequency encoding. And how does this work? So this technique, you're just going to replace the category with the respective frequency. All right. Now, feature selection, you must keep in mind after doing all this pre-processing, only specific features are going to contribute towards the prediction of the survival of a passenger. All right. So we want to see those features that contribute the most and those that contribute very little or don't contribute at all. So in this case, we're going to throw away all the features that don't contribute. So one of the methods is called variance threshold. And how does it work? So if you notice it, I said this thing, you eliminate the features with a very low variance. All right. Now, when I use the word very low, so you need to set a threshold. So some of the recommended variance that you can use is 3% to 5% for well-defined project. Now, when I say well-defined project, I'm looking at application. For example, I'm building this algorithm, which I'm going to deploy out there to be used by people, you understand? So I'll stick the variance to 3 to 5%. If it's just research, it's just for the sake of observation, so it's fully defined, you can use your variance between the interval of 10 to 15. All right. Again, they said a variance threshold estimator with the cutoff threshold. Remember what I mentioned about the cutoff threshold value? So it ensures that all features with variance below the threshold, they drop. All right. And then we said the features must be numerical. So before you apply variance threshold, we'll make sure that all your features are numerical. Hence the reason why we converted all categorical features to numerical values. All right. And then you must normalize, very important to ensure that the variants are on the same scale. There we go. So that is just a definition for sample variance. So everyone is familiar with this. And in terms of population variance, that's what you're going to do. So depending on the data set that you're working with, you want to use one over the other. So as a continuation, the next method I'm going to mention is lasso regression. All right. How does this work? So this technique selects features with a non-zero coefficient in filter regression with L1 regularization. All right, I'm just going to show a little image here to show how lasso loss actually looks like. If you notice there is a penalty parameter there. It's called alpha. So by adjusting the alpha value between 0 and 1, it actually enables some of the features to actually be tracked. All right. Yes, actually mentioned the penalty component of the loss function, which is the alpha that I mentioned, allows the coefficient values of some input variables to decrease to a value of 0, thus allows the associated features to be removed from the model. And very important, lasso stands for least absolute shrinkage and selection operator. What it does is adds an absolute value of magnitude. Now, if you look at the formula there in the penalty part, so the second piece of the formula, you have the penalty parameter alpha. If you look inside the sum, that's what they talk about, the absolute value of magnitude. All right. So what it's going to do is going to add this magnitude of coefficient as a penalty term to the loss function. And the hyperparameter called alpha, which I mentioned before, it controls the weighting of the penalty to the loss function. All right. So that's basically for the high level presentation. What's going to come next is actually the implementation. So in terms of the implementation, I've implemented this using Python, and I implemented all the methods so that we actually see how they work and how the output actually looks like. All right, thanks everyone. Is there any question before we move to the practical part?
Are there questions from the online people? I think Marcel wants to give a practical.
Will the practical be online or?
Yes, I can.
Okay, so if none, then you may also ask after Marcel gives a practical.
Yes, you can take the question after the practical session.
Can you start now? You can do it now. Because we have time, yeah. Because we have time. It's just 30 minutes. No, it's just 30 minutes. You can continue. Yeah, okay. Huh? Okay, so I think no. Yeah, sorry guys. So I think Marcel has to change laptop. So what we can do is we can. I mean, if there's any questions, you can ask now. Otherwise, we have to end early and then proceed with the practical after.
Just remind people of the presentation at 3 p.m. The first information on this.
All right, so just a reminder, in the chat, we put the online lecture for the afternoon. Okay, it's 3 p.m. South African time. So I know for the other countries, but just accordingly. So thank you all. So we'll see you in the afternoon. And for those here, okay, Marcel is gonna start the practical session. All right, thank you. But I'll leave it for 5 minutes or so if you have any questions. Okay.
So we're going to link up his laptop there.
No, I think he wants.
Yeah, Python is not.
No, there's no Python.
That's why if we link up your laptop there, then at least you can use the screen, right?
Yeah.
Yeah. Use your laptop.
Because I'm class, that's one.
So now we're going to move to the practical slide. I'm just going to implement the machine learning algorithms to the processing. All right. So I'm using Spider 3.8 with the other softwares that you can use. You can use Jupyter Notebook or any other platform that works well for you. So in Spider, what you typically want to do before running in the code is to make sure that your directory is properly set. So if I go to files, I'm just going to navigate to the correct location where I store my Python files. Right, so what you want to do is to say set console working directory. All right, without doing that, when you try to run the code, it's going to complete that directory not found. So as you can see there from the directory, I moved a step further. I did the pre-processing, but I also did a quick test to see how machine learning can predict and compare to the competition, how I perform compared to the others on the board, and it was really impressive. Okay, so the first thing I'm going to mention is the specific libraries that you're going to need. So you need a numerical Python library and you need pandas, right? So first I'm going to do is I'm going to import these two specific libraries. Those are my numerical Python, the pandas library. Now our data set is called train.csv. So I'm just going to read the CSS file. And then after reading the CSS file, the next thing I want to do is to get some information about the data that I just read. So data.info will tell me exactly how many variables are there and what are the data types associated with each variable or each feature. So there you can see, I don't know if everyone can see that. I'm just going to scope a little bit. You can see there data.info tells you that you have 12 features that you're working with. You have integers, floating values, as well as objects. So object is just to remind you that it's categorical. And then if you say data.head, it's just to give you a sample of the data. So it typically gives you first 5 records, right? You can see what the data actually looks like. So you clearly can see the integer values. And if you look at cabin there, there is stuff like C and 5, it tells you, and in back as well, it shows you that you dealing with categorical and numerical. All right. Now, what you want to do is you want to split this data. So you want to classify them into your input features. So those features which we're going to pre-process and we're going to fit into machine learning. And then your output levels or output feature is what we're trying to predict. So first I'm going to extract is what my output level is. So my output level, now if you notice it, Python is 0 index space. So what that means is it starts from 0, not 1. So if you check there on the little window there on the bottom right hand side, the second column called survive is actually labeled as index 1. So the passenger ID is index 0, survive is index 1. Hence, you see the reason why that code I'm highlighting. I'm saying that select all the data using index location or rows column 1. All right. So what happens there is I'm just going to show you using the variable explorer. The reason I choose to use this software specifically is because we can quickly see everything, which is a little bit difficult when you're using Jupyter Notebook. Right. So I'll just go to output level. There we go. So those are the output levels. You can see just 1 and 0. Tells you if a specific passenger actually survived the accident or not, right? So moving on now, I need to extract the input features. So these are the features that we're going to use, which we're going to pre-process we're going to feed into the machine learning, and then we're going to tell the machine learning that these are the output levels, take this input and try to predict, all right? So I'm also going to copy that. And every time you create a variable, for example, I call it input data, and you copy data, make sure you go and check to see that you copied everything. Because if you miss out one specific feature, then it changes your accuracy, right? Or it actually affects the performance of the model. Now, someone will probably ask me, why am I using? I'm listing all the columns. So if you notice it, you can read this in a range. It starts from 0, it skips 1, then moves from 2 all the way to 11. So in Python, you can just say 0, 2 to 11. It doesn't work, all right? So in this case, you need to list them out, all right? Then the next thing now, we need to count the number of missing data for each feature, right? So I'm going to say input data. I'm looking at the input data. I'm going to say it's not the sum. What this line does is it actually counts all the missing values for each of the features, and it actually shows us there. So I hope everyone can see that. So you see the passenger ID that is missing. We move down to age. We have 177 missing information. And then for cabin, there is a lot that's missing. There's 687, and back is just 2. All right. And the next one here is to actually tell us the balanceness of our data. We want to see, is this data balanced or not? How do you know the balanceness of the data? You focus on the output levels. You want to see how many times did a 1 appear compared to a 0, right? So that comparison will tell you if your data is actually balanced or not. All right. Keep in mind that we use all this input data to predict this output. So to know the balanceness, we don't actually need those input data. We focus on the output level, and we just check how many 1s and 0s are there. All right. So if you do that, you see that there are 549 zeros and 342 1s, which shows that our data is imbalanced. All right. You have 30-something% compared to about 60-something%. Right. Then the next step of pre-processing. Yes.
Okay. Please, in the case where the data is not balanced, so what is the solution to an unbalanced data?
All right, so if your data set is very large, then you can use a subset. All right, so you can actually try to balance it out by using a subset to such a point where there is a close relationship between 1s and 0.
In that case, your sample reduces.
Yes, so your sample reduces. So it's a trade-off. So it's a trade-off. Right? Now we have to handle the features that are irrelevant. So irrelevant features are those features that contribute absolutely 0. So they have no impact at all to predicting if a passenger is going to survive or not. And you can see there is actually 2. See, they are highlighted in green: passenger ID and name. Right? If you have a lot of those columns, you can just go ahead and list them out. And I am actually doing an in place. I say in place equals to true means that looking at this data set, drop these 2 features, but keep everything else. So if I run this line, I go to my input data. If you notice it, so input data, they changed. I don't know if you can see it on the top right-hand corner, where my cursor is now 891 records by 9 instead of by 11. All right. So it's supposed to be from 0 all the way to 10. Since I've already extracted the output level, so we initially had 11 input features. But after dropping those 2, we actually have 9. All right. So these other 2 empty matrices that I create here is just to store the column index that contains 70% missing value or more, and then the next empty array or matrix there stores rows that contain 100% of missing information. And the reason why we need to capture this information is because if you have a record that contains 100% missing data and you still have a prediction, it doesn't make sense, all right? Because you can't say, given no input, I actually have a prediction. That doesn't make sense. So all those records that contain entirely nulls or missing values, we have to clear them up, right? And like I said, in terms of the columns that contains 70% or more is based on research. I've seen most people, they choose to say, okay, my benchmark is 70%, especially if your data set is very big, then it's fine, like I mentioned at the beginning. If your data set is too small, you may still want to work with the 70%, right? So there I'm just going to execute this 2 lines just to create those empty arrays. And there you can see that in the comment, another good way of programming is to make sure you always have comments so that the next person picking up your code will be able to understand what you're trying to achieve. So yes, say finally delete all rows that contain entries of null. And this is extremely important in pre-processing for machine learning or topological data analysis because like I said before, if you have a row of 0 record that has nothing, then it will affect your performance at the end because you can't say for no input, I have a prediction. That doesn't make sense, right? So I'm just going to run the whole of this block. And what you're going to notice is, nothing changed if I move my cursor back up there. So top right hand corner, notice that input data, the size didn't change. You still have 891 records and you have 9 features. Nothing changed means that there were no rows that contain entirely missing data, all right? And then just for the purpose of this demonstration, I'm going to execute the next one that says that find and delete columns containing more than 70%, all right? So it's a strategy that I also use for my PhD because like I said, my data set was extremely large and leaving those data that contains 70% or more, I noticed that my accuracy dropped a lot. So I'm just going to apply the same idea here. So we're going to drop all those features that contain 70% or more, right? Like I said, the benchmark, you can choose to say 80% or 90. Then when you go to the end, you check your accuracy, you come back and adjust this. There's a rule of thumb. If you check most papers that are published out, they mention 70% or more. All right. Now, if you notice, if I move again, top right hand corner, see it changed there. Now I have 891 by 8. What does that mean? It means that one of those features actually dropped. And we know it's the cabin number. All right. So the cabin number actually dropped because there's a lot of missing information there. All right. The next step is feature engineering. Remember, feature engineering, we said this is where you want to use existing data to try and create new features for yourself. You can be innovative about this. There's no specific rule of thumb about feature engineering. All right. Looking at the data, you can think of something very innovative, come up with new features that can help your machine learning to become even smarter. So in this case, I'm thinking of 2 possibilities I mentioned before. It's just count the number of missing values for every record. And then I also indicate with the Boolean value, if for every specific record, there is a missing value or not, right? And before I execute that, I need to mention this. For feature engineering, these extra features that you added, if they're irrelevant, then by the time you get to the stage of feature selection, they get eliminated, all right? So if you add something here that doesn't contribute, then at the end of the day, it will still get removed. But I tried this for my PhD and one of these features got removed and the other actually stayed, showing that they actually contributed, which is actually cool. All right, so after running this, so if you check there the row NaN count, I'm just going to show you how my input data actually looks like now. All right, if you look at input data, my cursor is in the top right corner, notice that it changed. Initially, we had 8 features, now we have 10 to indicate that we've actually added those 2 features using feature engineering. And the cool thing about this platform is that I can actually show you how the data frame looks like at every stage of execution. So if you go there at the very end, you see that it adds something like a number of nulls and a Boolean value. I don't know if I would say it's quite fortunate or unfortunate that most of the time where there is a Boolean value, the number of missing values is also 1. So when you see a 1, it appears to be a 1. But keep in mind that for most other data set with missing value, this is not going to be the case. All right. But as long as you have a value on the number of missing data, there will be a 1 on the Boolean that says that there exists a missing value. All Now we move to handling of numerical features. All right, for the handling of numerical features, all right, what you want to do is a very smart way of handling these missing values in a very large data set. The cool part about Python is that they give us this library. So if you look at the scaling library, they give us a class called simple imputer class. What it does is it's able to look at your whole data frame, numerical, and then replace all missing values corresponding to a particular column with the mean of that column. So you don't have to write the code that says, check for column 1, check for column 2, and so on, understand? So the simple imputer class does all of this in one go, right? So what I'm going to firstly do is I need to extract all the columns that are numerical. I'm going to extract all the columns that are categorical. Why? Because I want to apply my simple imputer class on those columns that are numerical, right? So in this case, I'm going to apply it to all of them because when your data set is too large, it's hard to see if a specific column actually have missing value. Okay, obviously, if you write, if you actually run the previous code that I showed you, that say it's not the count, I actually tells you all of the columns that have missing values. But if you have to look through all of them, it's going to take a lot of time. So the best advice is just run your simple imputer class on all of your numerical data set. Why? Because if a column does not contain any missing value, it's just going to skip that. All right. So first I'm going to do is to extract the numerical data and the categorical data. And then here, like I mentioned before, I implemented everything that I mentioned in my presentation. So it's just for you to choose. So you can try the mean, you can do the median, you can do the most frequent, you can choose some constant values. So I've implemented all of them. But for now, we'll just probably do the first one that comes there, which is the mean, just for demonstration. Like I said, good practice is you don't want to stick to one method. So most people make that mistake of I'll replace my missing value with the mean. All right, how sure are you that the median is not going to give you a better result, or using most frequent is going to give you a better result? So try all of them. Like I mentioned, for the sake of demonstration, I'm just going to do the first one. Okay, I just needed to run import this library first. So from the scalar.input, I'm going to import our simple imputer class, and I'm going to run the code again. I'll just give you some time. All right. So there we go. So now to show that it actually works, if I go to numerical data, you'll notice it. So I'll just show you a quick demonstration, numerical data, to show you that it works. If you look at index number 1, so I'll use column 2, which is index number 1, it corresponds to age. You can see that there's someone's age there, which is 29.691. All right, that alone shows you that it actually worked, meaning that it computed the mean. So like I mentioned, you don't have to stick to mean. You can use the median, the mode, and so on. Right? In this case, we're just going to demonstrate the mean. Also, if you move to column 4, you can see that the second value there is also replaced by the mean. I think the fourth one as well, and so on. And it's always nice that when you do this thing, run through your data sets, scroll through and see if you can find any missing value. Don't assume that the code is perfect, right? Sometimes you write the code and let's say in the process of reading those numerical features, you actually miss one out. But if you make it a habit of always checking that things are working, then you might pick up all those errors. So there, if I scroll left, right, forward, backward, and so on, I don't see any missing value. The missing value, you're going to see something like 9. And the end that says not a number, all right? So in this case, we see that every missing value has been replaced with a mean, all right? So I'm just going to skip this other method because if I run them, there will be little or no effect since all our missing value for numerical features have been replaced already. Now we move to handling of categorical features. So missing values in categorical features, right? Here again, you have two possibilities. You can use the most frequent, or you can just use a constant text value or string value. So just for the sake of demonstration, I'm going to use the first one, which is the most frequent, right? So I'm going to run the most frequent. Again, if I go to categorical data, like I said, always check to see that things are working. Because if you don't check, you might have made a mistake, and at the end of the day, your machine learning is giving you very bad accuracy. You don't understand what. So you can see that if I scroll through this data, you see that all the missing values have actually been replaced by the most frequent, right? So you can see that all the values are there, so it shows you that we actually work. So I'm just going to skip the next method that's replaced with the constant value, since we've already done the most frequent method. Now we go to encoding of categorical data. Why do we need to encode categorical data? Because we saw machine learning models, they want numerical input. They don't want text. They want numerical input. So we need to encode it. And here also I've implemented one word encoder, dummy encoding, and the hash, right? Now I'm going to give you a little bit of insight before we're going to choose how you should go about it. For categorical encoding, you know already that is probably considered. So for a small data set, categorical encoding will work, where you might run into a problem called dummy variable trap, right? Dummy variable trap, of course, where you get multi-collinearity. So you have two or more features that can help predict another feature. You don't want that to happen. That's why you either stick to 2 or 3. So for 2, again, is dummy. So dummy uses k-1 bit, which is very important because that helps us solve that problem of dummy variable trap. And keep in mind that if your data set is too big, then it doubles in size, basically. It's going to be very huge. So if you already have 50,000 records, you're looking at more than 50,000 now, all right? And it also depends on the number of categories. So for example, if a feature is country, for example, let's say my categorical feature is country, and then you list 50 something there, and each of those needs to be represented using, so 50 minus 1, so 49 bits, you see that your data set is going to be used. And each of those representation is a new column on its own. So hence, even the dummy encoding doesn't work best for used data set. I've tried before with my data set page, it didn't work. My computer froze and nothing works. Oh, it's too big. All right, it doesn't work. Unless you have a supercomputer somewhere, then you can go ahead and do that. Now, hashing is the best option. We have very used data set. What hashing does is, by the way, hashing is one-way encoding. So what it does is it can represent maybe 50 features using a hash of size 10. Understand? But now the downside is, even though you can reduce the size of your hash, so by comparing the number of input variables to your hash size, notice that the hash size you can make it to be lower than the number of input variables. But the downside here is that you lose information, and also it becomes difficult to go back. When I say go back is, when I say one-way encryption, is that after hashing, you no longer know what is what. Understand? So your categorical features, all you're going to see is some binary value, some hash algorithm or some hash value, which you have no idea what is what. Understand? So if you that person that always want to keep track of which features, okay, this hash represent what features, then in that case, that can happen. So immediately implement hashing, there's no going back. It means you no longer know which feature is which. All right. So for our case here, we can choose to use either domain coding. Yes?
Yeah, thanks. I just wanted to ask, you said obviously it makes sense you're losing information during this. Do you have any idea or any control in terms of how much information is lost, or is it very much like you say, once you've gone down that road, you know?
Okay, in terms of control, I would say that, so it's just a rule of thumb also that I've seen from other machine learning guys are they always try and keep the hash size to be at least equal to or greater than the number of input variables. Because the minute you try to be greedy and you say make the hash size lower than the number of input variables, then you run a chance of losing information, and it even become dangerous because some of your features might use the same hash. Understand? Because the hash size is actually lower than the number of input. All right. So if you notice here, I use 644. I don't know if you can see there. So 644 here for the hash is because I've already done my homework. I've gone through the feature, so the specific categorical feature that has the highest number of categories. And if I remember well, this was for I can't really remember which one was that, but I actually did my homework. The highest number of categories that we got for any categorical feature was 644. So I make sure that I set it to 644, right? So like I said, it has to be 644 or more, all right? Because the minute you drop this number, they run a chance of using the same hash twice. And because of that ambiguity, the machine learning prediction accuracy also drops, all right? So I'm just going to run the dummy one here because remember our data set is very small. It's just 891 records. And if you check there for your categories, it's just 3. But again, must keep in mind that we have 644 categories for one of those features. Meaning after running this, the size is going to be very huge, right? I'm just going to run it so that you see what happens. Today wants it to be one-dimensional. Okay, I see what it's doing. So it wants me to say index location. I need to mention all of this location. Okay, just for the sake of demonstration, we're just going to leave that and we just stick to the hashing algorithm. Again, I'll explain why. Okay, it's because I didn't import this. Guys, before you quickly say that your code doesn't work, always read your error message. It says that it doesn't understand the attribute index locator, all right? What does that mean? It means I need to go back and check, am I missing anything? If you notice it, we jump into this without actually importing the categorical encoder class, all right? So the minute you try to run that code that uses that class, it's not going to work, right? So here, if you see that the first print, so this first print that hashing there is just to tell me that is to tell me how many categories are there, all right? And then the second print there was just to tell me how many categories. That's how I came to the point of noticing that it's 644. Like I said, for use data set, it's almost like a norm, you need to use hashing. So any of those other methods, the data set is going to grow exponentially used to a point where you can actually handle it, all right? All right, it's going to take a little bit of time because keep in mind, we have a category there that contains 644 levels. It means that it has to, it needs to generate a hash for each of those, right? And this usually takes a little bit of time. So this was super fast. From experience, I've waited sometimes for 2 hours or more. So this is super fast, all right? Now, this next step is to create a data frame. Remember, I have my numerical features with missing values. I replace those missing values by the mean. Like I mentioned for demonstrations, I use the mean. I had categorical features which have encoded. What you want to do at the end, you want to generate a single input data frame. So what do? You merge them up, all right? So this step is just merging everything up. So you can say I'm using pandas.concat. So I'm concatenating numerical and categorical. Why can I do that? Because everything is now numerical. And you can see the next 2 lines there, I'm adding those 2 extra features that were already numerical in nature, the one that says number of 9 and bold, right? So I'm just going to do that. I just needed to start by defining what this numerical. So I needed to convert it to data frame first. All right, very important before you can concatenate one data frame to the other, you must, or one array to the other, make sure that you convert it to data frame. So if you use pandas or concat, it means you're working with data frames, all right, not NumPy arrays. Very important. Hence, you see the first step there says convert NumPy array to data frame. So most people don't think the code is not working, nothing is happening simply because they're not following the rules. So arrays, you can't concatenate them using pandas or concat. Data frame, yes, you can do that if you specify the axis as 1. All right. So now I can run this code and everything should be fine. There we go. So everything looks fine. So new input data, let's go check how our new input data looks like. All right, it's very huge. So let me just show you from here. You can see here where I'm pointing. Look at the top right-hand corner. You see what's happening there? The number of records didn't change because remember we were focusing on encoding those features, which is column Y. Look how many features are there now, 653. We started with how many input features, 11. Now we have 653. And this is simply because using the hashing algorithm. If you use something like the one-hot encoding, trust me, you have like 10,000 already. All right, it will be crazy, right? It will be too much. So you need to watch out. So if you try to run an algorithm for pre-processing, your machine stops working. It's simply because your data has grown too large that your machine simply can't handle that. All right. So after explaining that, now we need to move to feature selection. So out of these 653 guys, very, very important because 653, they are columns. So it's considered as input features. Each of those columns goes in as a feature of its own. But not all of them can contribute towards the prediction. In fact, the majority of this will contribute 0% towards the prediction of a passenger dying or surviving, right? So you need to do feature selection. You want to select only those features that actually helps you or contributes to a significant level to predicting that a passenger is going to survive or not, right? So I'm going to start again by calling X to be my input features or input data. Y1 here, I call it my output levels. All right, so I'm just creating new variables just to store them. Now you have 3 options here. The first one is variance threshold. The next one here is lasso regression, which is we tune hyperparameters. And then the other one is still lasso regression, but this time the difference between the second lasso and the third lasso is that the first lasso uses something called lasso CV. All right, it uses lasso CV. Guys, to be very honest, lasso CV, if you've played with this before, it takes forever to run. And for every parameter that you use, for example, if you look at this value that I'm highlighting here, so I'm choosing my alpha value, which is from 0 to 1 in steps of 0.01. What it's going to do is it's going to split this infinite number of times, right? So it's going to take 0.01, run all the features, which you can see 653 is going to take a lot of time, right? So the best way to actually do this, okay, based on the data that I've worked with before, is using a grid search CV, right? Now, if you've done anything called hyperparameter tuning, whether it's in machine learning or anywhere, ANS, CNN, or any machine learning that you've done, you notice that they mostly use something called grid search CV. Why? Because it actually computes stuff very fast, and it actually slows down your computational time. So it gives you better accuracy, and at the same time, it reduces your computational complexity. So in this case, I'm going to stick to grid search CV, but for you to see that it works better, we go home, try and implement lasso CV and see what the difference is. Besides the long time that it's going to take, you notice that your accuracy will also be very poor, right? So here I'm just going to run the grid search CV. Oh, also I need to mention the variance. I skipped the variance. I was focused on the lasso regression. Now, the variance threshold method is it's a very nice method. Like I said, for well-defined project, you stick your threshold to be from plus or minus 3 to 5%. You can see that's 5%. Most papers out there, they use 5% because, like I said, between 3 and 5%. However, even at 5%, if you notice it is going to let through a lot of features that don't actually contribute. So I've implemented this so many times using very huge data set, and I noticed that my data set size almost stayed the same, and the accuracy was very poor. So the reason I'm saying so, it requires the variance of that feature to be lower than 0.05 for that feature to be removed, which in most cases is not going to happen, right? So rather stick to lasso regression, right? If you want good accuracy, stick to lasso regression. And again, another cool thing about lasso, why I'm recommending lasso is because you can actually tune your high parameters. So for example, the alpha value here, which is called the penalty term, or the penalty term, you can actually tune it. Instead of saying that use alpha equals to 0.01, which is what most places or most papers they use, you can actually create a grid CV or a form of a set space, understand, say that, okay, for every alpha value, between 0 and 1 to 0.01, run this and tell me which of these alpha value actually gives me the best possible result, all right? And in this case, if I run this whole block here, see what happens, it's going to tell me exactly what alpha value works best, all right? And remember, like I mentioned, you don't want to use fixed values. You want your algorithm to tell you which value is the best. Hence, you do hyperparameter tuning always, all right? So we if your alpha value is 0, you're not doing any form of regression. It means you're taking away the penalty term. And if you set it to 1, it means you're giving maximum penalty. So alpha set to 1 typically is going to take off all your features, right? If you try that, you notice after running your features, then you probably get like 1 or 2, right? So let's see how this works. Let's just give it a little bit of time. It's still running. Okay, just to mention, for those of you using Python for machine learning, for you to see how long, because I did that for my page and I had to wait for 1 hour, 2 hours, nothing is happening. So you see the step here where they say CV equals to repeated 4. Inside there, you can add an extra term. It's called variables, all right? Variables, always use variables. Set it to 2 or 1, so 1 or 2. So what that does is, it will tell you, it will give you an update. How long has it been running? How long is it gonna take, or how long left, and so on, understand? Because if not, you're gonna stay there for like 30 minutes or 1 hour, or even the whole day. So in this case, we didn't use variables, and we're lucky because the data size is not that big. You can see that it recommended an alpha value, top right, bottom. You can see there's the alpha, the recommended alpha that is 0.01. Like I said, these are not values that you can just guess. You need to you need to do a grid CV search to actually get the best alpha value. Now we're going to use this alpha value to select our features, right? So grid search CV is to find hyperparameters for your lasso regression. After getting that hyperparameter, that best parameter that you want, you go back to your lasso regression and then use that best parameter and then run your lasso regression that will actually extract those features, all right? So there is 0.01. You can see I've already put it because I tried this already, and it worked. So before I run this, there's just something that I need to mention quickly. So remember that we started before doing feature selection. So if you look at where my cursor is up there, your data frame is very huge. So you have 653 features, right? Which I can guarantee most of them, we call them noise. They don't contribute in any way. Now, after running this, you're going to see how many features going to the left. Okay, sorry for that. I just keep an import there at the top. It's called the train test split. So I just need to run that train test split. So what the class does is it automatically, if you set your test size to 20%, you automatically allocate your training size to 80%, right? So using this, you can actually specify what the size of your training set is and your test set, all right? So after doing that, now let's run this so that we can see how many features will be left out of 653. Let's see what is going to be remaining. All right, so let's find, if you notice it here at the bottom, I call it Titanic input data, just so that we can spot it nicely. Let's find Titanic input data, and let's see what the size is. All right. Now, if you look at this, so let me just open it. That is all that is basically left. Out of 653, we only left with 7 features. Right. So you see, after running all these complex encoding schemes, the end of the day, you're left with 7 features. But bear in mind that these 7 features, they're not features that you actually know. All right. You don't actually know what, for example, this column here stands for, column 350. Like I said, if you use hash encoding, you can't tell anymore which features which, right? So it's actually you leave this to your algorithm to actually tell you what actually works best. But like I said, the surprising part of it is that you will not be able to tell after using hashing algorithm what features which. At the end of the day, you want a good prediction, not actually to be able to tell what features which are. So that is what we basically left with. And the next line here is just to store it in some file. So I just create a file name and I'm storing it automatically somewhere. So let me just navigate to make sure, okay, I don't need to navigate. I can already see from, my directory. You see why it's always nice to set up your directory, very nice. So that you can see which file is and which file is not. Now, if you look at that direction in the top right, that file doesn't exist, right? So if it doesn't exist, it creates it automatically. That is the beauty. So if you specify a file name in Python, and in Python, you can see that it will create it, right? All right, and the reason why after pre-processing is stored in a separate file, you see that called Titanic tune dataset. Initially, we had a Titanic dataset that was raw data. After tuning, we have the Titanic tune dataset. Now, this file is what you're going to use for a machine learning algorithm to do some prediction. All right. Luckily enough for you guys, I have 3 machine learning algorithms already implemented. It's just a matter of clicking the button and running them, so that we see what the accuracy looks like. For the sake of time, I will just run one of them. This is maybe KNN. We can see choose any of them, right? So KNN, K nearest neighbor. How does work? I'm using default parameters. So test size of 20% and training size of 80%. Metric that's Minkowski, P equals to 2. So P parameter down is just telling me it's going to, so you using that nearest neighbor, what kind of distant metric is it going to use? Euclidean, is it going to use another distant metric? So I'm using 2. And then the default n neighbors is 5. So this is just default parameter. We just want to see how this algorithm is going to work. And for it to work, as usual, just make sure you always have your numerical pointer and pandas always ready, because if not, you will not be able to work with arrays or pandas. All right, you wouldn't be able to read any service file or work with arrays. So after doing that, I'll just read. Remember, I stored it as Titanic tune data set. I can read it. Then I can split between input and output. So what goes in an input feature and what am I trying to predict? So X and Y. And then again, I use my test train split class to split 20% test size, 80% training size. And then here, okay, very important, you need to do something called future scaling. So machine learning, always make sure that you scale out your information. You can something called normalization. So do feature scaling. Again, Python gives you good, libraries actually does it. So from scaling the pre-processing, there's a class called standard scaler. What it does, it will make sure that everything is actually on the same scale, or your input data is on the same scale. Just make sure that you scale out your data. And here, like I said, I'm using default parameters. So n neighbors, so just 5 neighbors. And then my distance, I'm using p equals to 2. If I remember, was actually Euclidean distance. And then I just need to fit my training set to the output that I'm trying to predict. Okay, that worked nicely. And then why predicted is just now I'm going to use that X test that I kept. Remember, I split it 80-20. I use it on at 20% to run the predictions. So just need to run this line. All right, so I've already done the prediction. But if you after running your prediction, what you want to do is you create something called a confusion matrix. So I'll show you exactly what confusion matrix is. Again, you need a class for that called confusion matrix. It's coming from scalar.matrix. So I generate confusion matrix. I'll show you now. So my advice is, if you have a confusion matrix, just make sure you add this extra line that says sum the trace over the total number of observations. Because if you don't do that, you want to rely on using a calculator to compute the accuracy. It's going to be a very bad idea. All right, that's what your confusion matrix is. You can see that so 0, 0, there's 100, 0, 1, there's only 10. So 10 times we got someone who died, or people who died, and they actually predicted to be alive. And then 19 times there were people that are alive that are predicted to be dead. So you can see already you can compute your accuracy from here. But I'll save you time and I'll do the code computationally. So there you just say confusion matrix the trace divided by the sum, and it will give you an accuracy, so 83.8%. And this accuracy, keep in mind, this is using default machine learning parameters. You can get it to 90%, 100% if you do something called machine learning hyperparameter tuning. All right. So this accuracy, you want to know how bad it is? The website I got a data set is called kaggle.com. They run competition. This data keeps you in the top 185, I remember well, and the total number of competitors is 13,000. So it keeps you in the top 0.something%. All right, means this result is good, even though in the machine learning where we know that we haven't yet done any tuning yet. All right. So if I have to get this to be better, I'll do some TDA on my data. I'll do some hyperparameter tuning, and I can push this all the way to 90-something%, possibly even 100. All right. That is basically it. There's no need for me to run the other machine learning algorithm. Okay, and then we can see that we have a very good accuracy. And that's basically for my presentation. Is there any question?
Okay, thank you, everyone. So for those in person who have questions to myself, we can discuss it over lunch. Okay? But for those online, okay, thank you for joining us for today. Just a reminder for this afternoon, again, it's at 3 p.m. South African time. So just calculate what time it is in your place. Okay. Thank you for joining us today, and hope to see you this afternoon and for the next coming days of the workshop. Bye-bye.